Welcome to this tranquil exploration of genetic engineering, a field that gently invites us to ponder the very fabric of life itself. As you settle into a state of calm, allow your mind to drift through the intricate world where science meets the subtle dance of nature. Tonight, we will softly uncover the principles and possibilities that lie within the modification of living things, moving at a pace that encourages deep relaxation and a quiet understanding. Genetic engineering is a fascinating area of study, touching upon our deepest questions about life, its origins, and its potential future. It is about understanding the detailed instructions that guide every living organism, from the smallest bacterium to the most complex human being. Imagine these instructions as a delicate script, written in the language of DNA, guiding development and function. Genetic engineering, in its essence, involves reading, understanding, and sometimes, gently editing this script. It is a profound interaction with the fundamental components of life, opening doors to both wondrous innovation and careful contemplation. As we journey through this topic, we will also consider the deep ethical pathways that accompany such powerful capabilities. Like a quiet stream flowing through a serene landscape, the ethical considerations wind alongside every scientific discovery, guiding our thoughts toward responsible and thoughtful progress. We will explore how societies and scientists have grappled with the implications of altering life, developing frameworks to ensure that respect, well-being, and justice remain at the heart of every endeavor. Our exploration begins with a gentle look into the historical moments that shaped our understanding of ethical conduct in research, particularly involving human subjects. These moments, though perhaps challenging, laid down foundational principles that continue to guide all scientific pursuits, including genetic engineering. They remind us of the immense responsibility that comes with the power to intervene in biological systems. Let us softly consider a time when the world faced profound questions about human experimentation. On a cold December day in 1946, German physicians were brought to trial for experiments conducted on concentration camp prisoners, experiments carried out without any form of consent. This grim event served as a stark, somber lesson, highlighting the absolute necessity of protecting human dignity and autonomy in scientific research. In the aftermath of these trials, a crucial document known as the Nuremberg Code was established in 1948. This code emerged as the very first international declaration to champion the idea of voluntary participation and informed consent for human subjects in medical experimentation. It was a profound step, articulating principles that are now considered fundamental. The Nuremberg Code gently reinforced that the voluntary consent of human subjects is not merely important, but absolutely essential. It also laid down a guiding principle that the benefits of any research must always clearly outweigh the potential risks to a person's health. These quiet pronouncements became a beacon, illuminating the path forward for ethical research around the globe. Continuing this journey through time, we arrive at another significant milestone, the Declaration of Helsinki. In 1964, the World Medical Association, a global body of medical professionals, released a set of recommendations designed to guide doctors involved in medical research. This declaration broadened and deepened the principles established by the Nuremberg Code, providing a more comprehensive framework for international research ethics. It specifically outlined rules for different types of research, including studies that combined clinical care with research, and those considered purely non-therapeutic. Today, the Declaration of Helsinki serves as the very basis for what are known as good clinical practices, which are universally adopted standards ensuring the ethical conduct of clinical trials. The Declaration of Helsinki gently addressed several key issues to ensure the well-being of research participants. It stipulated that any research involving humans should be built upon a solid foundation of prior laboratory and animal experimentation, ensuring a careful, stepwise approach. Furthermore, it emphasized the vital role of an independent committee, which must review all research plans before any study begins, providing an impartial layer of ethical scrutiny. Informed consent, a cornerstone principle, was reaffirmed as absolutely necessary from all research participants, ensuring that individuals fully understand and willingly agree to their involvement. The declaration also underscored that research should only be conducted by individuals who are medically and scientifically qualified, preventing unqualified individuals from undertaking sensitive human studies. And perhaps most importantly, it reiterated that the risks of any research must never, under any circumstances, exceed the potential benefits, placing the safety and well-being of the human subject above all else. However, the path to establishing robust ethical guidelines was not without its shadows. The Tuskegee syphilis study, a long and deeply troubling chapter in medical history, serves as another powerful reminder of the consequences when ethical principles are disregarded. This study, conducted by the U.S. Public Health Service from 1932 to 1972, involved 600 low-income African American men. Among them, 400 were positive for syphilis, and 200 were negative, serving as a control group. Participants were offered what were described as free medical examinations, but they were never informed of their actual disease status. As the decades passed, a cure for syphilis, penicillin, became widely available in the 1950s. Yet, the participants in the Tuskegee study were tragically denied this treatment. Even more distressing, in instances where physicians not involved in the study diagnosed subjects with syphilis, researchers intervened to prevent those individuals from receiving medical treatment. This deliberate withholding of life-saving care led to immense suffering and avoidable deaths. The Tuskegee syphilis study provoked overwhelming academic and public outrage, eventually leading to its termination. It marked a critical turning point, spurring a broad consensus on ethical guidelines for biomedical research and directly leading to the formation of the National Commission for the Protection of Human Subjects of Biomedical Research in the United States. This commission was tasked with ensuring such a grave breach of ethics would never happen again, moving society toward a more compassionate and just approach to scientific inquiry. Building upon these difficult but instructive historical experiences, the Belmont Report was published in 1979 by the National Commission for the Protection of Human Subjects of Biomedical Research. This pivotal document was designed to summarize the basic ethical principles identified by the committee and to provide clear guidelines for resolving ethical problems encountered in human biomedical research. It distilled the complex landscape of research ethics into three core, overarching principles, offering a foundational framework for responsible scientific practice. These three basic ethical principles are respect for persons, beneficence, and justice. Respect for persons acknowledges the autonomy of individuals, recognizing their right to make their own decisions. It also includes protecting those with diminished autonomy, such as children or individuals with cognitive impairments, ensuring they are not exploited. Beneficence is the ethical obligation to do no harm and to maximize possible benefits while minimizing possible harms. It encourages researchers to constantly evaluate the risk-benefit ratio of their studies. Justice concerns fairness in the distribution of research benefits and burdens. It means that research subjects should not be chosen merely because they are readily available or vulnerable, but rather because they are relevant to the research question and that the benefits of the research should be accessible to all who could benefit, not just a select few. These three principles, interwoven and interconnected, provide a guiding light for navigating the complex ethical terrain of human research. In the United States, these principles were further codified into what is known as the Common Rule, a set of federal regulations that govern ethical human biomedical research. The Common Rule laid out specific requirements to ensure that research institutions comply with ethical guidelines. It detailed the necessary steps for researchers to obtain and meticulously document informed consent from participants, ensuring transparency and voluntary involvement. It also established requirements for the membership, function, operations, and review processes of institutional review boards, or IRBs, which are independent committees responsible for evaluating research proposals. These boards play a critical role in safeguarding the rights and welfare of human subjects. Furthermore, the Common Rule provided additional protections for particularly vulnerable groups, such as pregnant women, prisoners, and children, recognizing their unique needs and ensuring they are not subjected to undue risk or exploitation. These detailed regulations create a structured system of oversight, striving to uphold the highest ethical standards in all human research. Beyond the specific regulations of the Common Rule, broader systems of oversight have been developed to ensure ethical research practices across different countries. At the heart of these systems is the Research Ethics Committee, an independent body responsible for reviewing research proposals before they can begin. These committees ensure that all research adheres to legally binding standards, safeguarding the rights and well-being of participants. their primary role is to ensure the protection of those who volunteer to be part of scientific studies. In both Canada and the United States, the institutions that conduct research also bear a significant responsibility for its ethical review. Most academic institutions, for example, have their own internal research committees dedicated to this task. In Canada, a specific body known as the Research Ethics Board, or REB, operates under the guidance of the Public Health Agency of Canada. Similar research models are followed in many other developed countries, reflecting a global commitment to ethical science. In Europe, a regional ethics committee often reviews all research within a given geographical area, creating a consistent standard across a broader region. These layers of oversight, both institutional and national, work together to provide a comprehensive safety net for human research, ensuring that scientific progress is always balanced with profound ethical care. Canada, in particular, has developed a robust system of oversight, with 3 federal granting agencies playing a key role in ensuring ethical compliance. These agencies include the Canadian Institutes of Health Research, the Social Sciences and Humanities Research Council, and the Natural Sciences and Research Council of Canada. Any institution that receives funding from one of these agencies is legally obligated to ensure its research adheres to the Tri-Council policy statement. This comprehensive policy serves as the primary source of concrete guidance for the oversight of human research in Canada. Additionally, Health Canada, the federal department responsible for national public health, generally requires sponsors of clinical trials for drugs and biomedical devices to obtain approval from a research ethics board before proceeding. This ensures an additional layer of scrutiny for interventions that directly impact human health. The Tri-Council policy statement, first established in 1998, is intended to be a living, evolving document. This means it is regularly reviewed and renewed annually to reflect new developments in research and ethics. Over the years, the updated statement has significantly grown in length, nearly doubling its original size, and has considerably improved upon its initial guidance. Both Canada and the United States, despite their different names for regulatory bodies, have adopted similar systems of ethical review, often grappling with comparable challenges. The increased length of the Tri-Council policy statement allows for a broader range of issues to be addressed, extending beyond the scope of the common rule in the US. This includes offering further guidance on sensitive areas such as research involving Indigenous peoples, managing incidental findings that are relevant to a participant's health, addressing potential conflicts of interest, and navigating research conducted during emergencies. This continuous evolution ensures that ethical frameworks remain relevant and responsive to the complexities of modern scientific inquiry. The core principles of the Tri-Council policy statement are a gentle echo of the foundational ethical principles we discussed earlier. They are respect for persons, concern for welfare, and justice. These overarching principles are not confined solely to research ethics but extend to broader ethical considerations in general. It's important to understand that none of these principles can be simply absorbed into another or given automatic priority over the others. For example, the principle of respect for persons dictates that you must respect an individual's autonomous decisions, even if those decisions might seem to affect their welfare in a way that others might view as less than optimal. This delicate balance requires thoughtful consideration in every ethical dilemma, ensuring that all aspects of human dignity and well-being are carefully weighed. However, even with robust systems, there can be pitfalls in institutional review. One potential advantage of having ethics committees at the institutional level is that their members are often more familiar with the specific research being reviewed, given their proximity to the ongoing work. This familiarity can streamline the review process and make it easier to monitor outgoing research for compliance. It also makes it possible to impose institutional sanctions on investigators who violate established policies, providing a clear mechanism for accountability. Yet, institutional review also presents certain disadvantages. A significant concern is the increased likelihood of conflicts of interest, especially when investigators on the ethics committee are also colleagues with those whose research they are reviewing. This can subtly influence decisions. Furthermore, modern research increasingly involves multiple institutions and often spans international boundaries. In such multi-site, international endeavors, a separate research committee at each site must review the same research proposal. This can lead to a considerable consumption of time and resources as researchers navigate multiple review processes. Adding to this complexity, researchers may encounter inconsistent judgments from various ethics committees, creating confusion and delays. These challenges highlight the ongoing need for harmonization and efficiency in ethical oversight, ensuring that necessary scrutiny does not inadvertently impede valuable scientific progress. At specific institutions, like McMaster University, the system of ethics review is tailored to manage the diverse range of research projects. Generally, if a principal investigator, or PI, is appointed by any faculty other than the Faculty of Health Sciences, their study is reviewed by the McMaster Research Ethics Boards, known as MREB. However, if the principal investigator is from the Faculty of Health Sciences, Hamilton Health Sciences, or St. Joseph's Healthcare Hamilton, then their project must be reviewed by the Hamilton Integrated Research Ethics Board, or HIREB. For student research, if both the faculty supervisor and the student investigator are from a faculty other than the Faculty of Health Sciences, the study is reviewed by MREB. But if both are from the Faculty of Health Sciences, then the study falls under the purview of HIREB. This differentiated approach aims to ensure that studies are reviewed by boards with the most relevant expertise and understanding of the specific research context. The Hamilton Integrated Research Ethics Board, or HIREB, is a crucial entity responsible for ensuring that all human research projects involving McMaster University, St. Joseph's Healthcare Hamilton, and Hamilton Health Sciences obtain ethical approval before they can commence. HIREB's core responsibility is to ensure that all human research adheres to the highest ethical standards. Beyond just approval, it also provides valuable advice on the ethical, scientific, and technical aspects of planning research projects. The HIREB Executive Committee has specific duties, including reviewing and approving minimal-risk research projects that meet acceptable ethical and scientific standards and have adequate facilities and resources. They also oversee serious adverse event reports, protocol deviations, violations, waivers, and annual progress reports of approved projects, with these activities being reported to the full HIREB. Additionally, specialized committees exist, such as the Human Tissue Committee, which reviews studies solely focused on the collection and analysis of human tissue, and a Student Research Committee, ensuring tailored ethical oversight for all types of investigations. In an ongoing effort to refine ethical guidelines, the Tri-Council policy statement has recognized areas where its initial scope needed expansion. For instance, the original policy statement, designed around a biomedical model, was never specifically intended to cover non-biomedical research, such as studies involving intervention or interaction with living things in the social sciences. This led to some social science researchers expressing concerns that a biomedical model was being inappropriately imposed on their discipline, potentially stifling important and innovative research in those fields. Recognizing this, the updated statement places a greater emphasis on distinguishing between the different standards that should apply to various types of research. For example, separate chapters have been developed specifically to discuss qualitative research and clinical trials, acknowledging their unique methodologies and ethical considerations. This more nuanced approach is the result of an extensive process of consultation and collaboration with academics in the social and behavioral sciences, ensuring that the ethical framework is both comprehensive and appropriately flexible for all disciplines. The evolving Tri-Council policy statement in Canada continues to place significant emphasis on flexibility, more so than its US equivalent, the Common Rule. This updated statement consistently promotes a proportionate approach to research ethics review. This means that research deemed to be most risky receives the most intensive scrutiny, time, and resources, while less risky research may undergo a more streamlined review process. This allows for a more efficient allocation of limited resources within the ethics review system. Individual institutions are granted considerable leeway in deciding how to implement this proportionate approach, including determining who may conduct delegated reviews and how to structure their internal processes. This flexibility serves as a valuable model for others to consider, as it encourages Canadian institutions to maximize their ethics review resources by focusing them on the research that is most likely to raise significant ethical concerns, thereby promoting both scientific integrity and public trust. Now, let us gently shift our focus from the broad ethical foundations to specific case studies and the profound power of synthetic biology, a field that deeply intertwines with genetic engineering. We will consider recent events that highlight both the immense promise and the serious ethical challenges of altering the very code of life. Consider the CRISPR baby scandal of 2018, a case that sent ripples of international concern through the scientific community and beyond. On November 26, Dr. He Jiankui, an expert in DNA sequencing at the Southern University of Science and Technology in Shenzhen, China, made a startling announcement. He declared that he had disabled a specific gene, CCR5, in human embryos. This gene is known to be involved in helping HIV enter healthy cells. The result of his work, he claimed, was the birth of twin girls, named Lulu and Nana, who were resistant to HIV in early October of that year. This announcement immediately sparked an international outcry. The scientific and ethical implications were immense and widely condemned. Dr. He Jiankui was subsequently sentenced to 3 years in prison for violating government bans on the clinical procedures of gene editing in human embryos for reproductive purposes. He was also fined 3 million yuan. In response to this deeply troubling event, the Chinese government significantly tightened restrictions on its human genome editing policies. This case underscored the critical importance of global ethical consensus and strict regulatory oversight when applying powerful genetic technologies, particularly those that could lead to permanent, inheritable changes in the human genome. It serves as a potent reminder of the delicate balance between scientific advancement and profound societal responsibility. In contrast to the ethical controversies, CRISPR therapies also present immense hope for treating debilitating diseases, as illustrated by ongoing experimental studies. Currently, there are about 50 experimental gene editing studies underway globally, with 40 of them utilizing CRISPR technology. More than 200 people have already been treated with these experimental therapies. A notable success story comes from Vertex Pharmaceuticals, which reported curing more than 75 people of sickle cell disease. Sickle cell disease is a painful and debilitating condition caused by misshapen red blood cells that cannot carry enough oxygen, leading to severe pain and anemia. The challenge in these therapies, however, often lies in efficiently delivering CRISPR to the specific cells in the body where it needs to act. In one remarkable case, that of Victoria Gray, doctors had to remove bone marrow cells, edit them using CRISPR, and then, after chemotherapy to eliminate her remaining bone marrow, reintroduce the newly edited cells to make room for them to thrive. While these are still experimental therapies, the first ones are expected to receive approval soon, heralding a new era of genetic medicine. This rapid progress showcases the incredible potential of genetic engineering to profoundly improve human health, offering a beacon of hope for previously untreatable conditions. The power of synthetic biology extends far beyond individual gene edits, encompassing a broader vision of designing and building new biological systems. Synthetic biology is that branch of research and development that applies rational design principles to produce novel biological systems, organisms, or components from scratch, or de novo. It also contributes significantly to their creation through the novel development of materials, technologies, or processes. Imagine a methodical approach, much like engineering, but applied to the living world. This involves integrating biology, engineering, and informatics to move from rational design to the creation of DNA, proteins, and cells, ultimately leading to high-value applications. A key aspect of this field is the development of standardized biological parts or modules, often referred to as biobricks, which are open-source components that can be tacked onto bacterial chassis to produce customized biological systems. This modular approach allows scientists to construct complex biological circuits with greater predictability and ease, much like building with Lego bricks. At the forefront of synthetic biology's capabilities is CRISPR technology, often pronounced CRISPR. This acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats. These unique genetic sequences are the hallmark of a bacterial defense system, a natural mechanism that bacteria use to protect themselves from invading viruses. This ancient bacterial defense system has been cleverly repurposed to form the basis for CRISPR-Cas9 genome editing technology, a revolutionary tool in genetic engineering. CRISPR systems can be precisely programmed to target specific stretches of genetic code within a cell, allowing scientists to edit DNA at exact locations. With these remarkable systems, researchers gain the ability to permanently modify genes in living cells and organisms. Looking to the future, this technology holds the promise of correcting genetic mutations at precise locations within the genome, potentially offering cures for a wide range of inherited diseases. The precision and versatility of CRISPR have made it a cornerstone of modern genetic engineering, opening up unprecedented avenues for understanding and altering life. The journey from synthetic genes to synthetic viruses and even synthetic bacteria highlights the rapid advancements in our ability to engineer life. The first full gene was chemically synthesized in 1972, a groundbreaking achievement that demonstrated our capacity to build genetic material from its basic components. This initial step paved the way for more complex creations. Fast forward to 2002, when Eckhard Wimmer and his collaborators reported the successful synthesis of the 7.5 kilobase poliovirus genome. This was a significant leap, taking approximately a year to accomplish. Just 1 year later, in 2003, Craig Venter's group announced that they had synthesized the slightly smaller X174 bacteriophage virus in a mere matter of weeks. These achievements were not just about creating copies, they demonstrated the ability to construct functional viral genomes from scratch, piece by piece. This progression showcased a growing mastery over the fundamental building blocks of life, hinting at the profound possibilities and responsibilities that lay ahead in synthetic biology. The ability to synthesize viruses soon led to even more ambitious goals, the creation of synthetic bacterial genomes. In early 2008, Craig Venter's group achieved another remarkable feat, succeeding in synthesizing a full bacterial genome. This was followed by an even more profound announcement in May 2010, the creation of the first living and replicating bacterium with a synthetic genome. This was not merely a copy of an existing bacterium, it was a cell whose entire genetic material had been designed and constructed in the lab. This synthetic bacterium differed only slightly from its wild-type counterpart, Mycoplasma mycoides, yet its genome was entirely man-made. This achievement marked a pivotal moment, as it represented the creation of bacteria that had not and indeed could not have naturally existed in nature. It demonstrated a profound capability to fundamentally redesign life, moving beyond simply modifying existing organisms to actually constructing them from a synthetic blueprint. This achievement further intensified discussions around the ethical implications of playing God and the very definition of life itself. This fundamental redesign of life continues to push boundaries. Floyd Romsberg and his colleagues have successfully developed 2 new, artificial DNA bases, expanding the genetic alphabet beyond the traditional 4. What's truly remarkable is that these new bases can be incorporated into DNA alongside the existing 4 and can even be replicated by naturally occurring enzymes. While the full potential applications of these new bases remain unclear, this breakthrough suggests that we may soon be able to create DNA with highly unusual characteristics, leading to entirely new forms of genetic information storage and function. Further exploring this concept, the PACE, or Programmable Artificial Cell Evolution, consortium has taken initial steps toward developing lifelike protocells. These protocells use peptide nucleic acid, or PNA, rather than DNA, as their information-storing molecule. This represents a foundational exploration into alternative genetic systems, hinting at the possibility of creating entirely new forms of life with different biochemical underpinnings. These advancements raise profound questions. Are we merely tinkering with life, or are we truly on the verge of fundamentally redesigning it? The implications for medicine, biotechnology, and our understanding of life itself are vast and continue to unfold. The high-value applications emerging from genetic engineering and synthetic biology are vast and deeply impactful. These fields are driving the creation of new drugs, developing more effective vaccines, and pioneering advanced diagnostic technologies. Imagine a future where diseases are not just treated, but preempted and precisely targeted. One exciting area involves synthetic agents designed as targeted cancer treatments. This includes the development of cancer-targeting viruses and novel treatments for HIV. It has been observed, for example, that certain wild-type bacteria naturally colonize tumors and exert an anti-cancer effect. Through synthetic techniques, researchers envision creating engineered agents that can localize to and attack cancer cells even more selectively, minimizing harm to healthy tissues. This precise, intelligent approach to medicine holds tremendous promise for improving outcomes for patients battling these challenging diseases, moving us closer to more effective and less invasive therapies. Beyond medicine, synthetic microbes are being harnessed to manufacture a diverse range of products. They could be used to create new drugs or other chemicals with unprecedented efficiency and specificity. For example, the company DuPont is already employing semi-synthetic bacteria to convert corn sugar into a chemical, which it then uses to produce various fabrics. This innovative application demonstrates how biological systems can be reprogrammed to serve industrial needs, offering more sustainable and environmentally friendly manufacturing processes. Several other companies are actively researching the possibility of utilizing synthetic agents to generate biofuels. These efforts aim to create renewable energy sources by engineering microbes to produce fuels from readily available resources, reducing our reliance on fossil fuels. These examples highlight the broad utility of synthetic biology, transforming not just healthcare but also industrial production and energy generation, paving the way for a more sustainable and technologically advanced future. The applications of synthetic microbes extend even further, venturing into environmental protection and novel technologies. These engineered organisms could be used to detect environmental toxins, acting as tiny biological sensors. Imagine semi-synthetic microbes capable of detecting subtle changes, like the presence of explosives, or even differentiating between the complex chemical profiles of espresso, coffee, and decaf. Such precise biological detectors could revolutionize various industries, from security to quality control. Another significant application is the deactivation of environmental toxins or pollutants, including the daunting task of mopping up oil spills. Researchers at institutions like Berkeley have already engineered a semi-synthetic bacterium that can effectively degrade the insecticide paraoxin, showcasing the potential for biological solutions to environmental challenges. Synthetic biology also opens doors to the development of biological cameras, computers, or even robots, envisioning a future where living systems perform computational and mechanical tasks. Furthermore, this innovative field has even found its way into synthetic art, where biological processes are harnessed to create unique and dynamic artistic expressions, blurring the lines between science, technology, and creativity. However, with such profound power come significant ethical concerns, which bioethicists carefully scrutinize. These concerns can be broadly categorized into several areas. First, there are general concerns regarding laboratory biosafety, ensuring that the creation and manipulation of novel biological systems do not pose unintended risks to human health or the environment. Second, there is the potential for synthetic biology to aggravate existing injustices, perhaps by making advanced therapies available only to a select few or by creating new forms of inequality. Third, the rapid advancements in synthetic biology pose challenges to existing systems of intellectual property rights, as the very definition of creation in a biological context becomes increasingly complex. Beyond these general worries, bioethicists have identified three major specific concerns. The first revolves around the idea of playing God or toying with nature. This concern has been prominent in closely related areas of science and questions whether humans are overstepping their natural limitations by designing and creating life. The second concern delves into whether synthetic biology might undermine the distinction between living things and machines. This issue attracted early attention from ethicists, questioning how we define life and whether engineered organisms retain the moral status traditionally accorded to living beings. Finally, the third major concern addresses the deliberate misuse of knowledge from synthetic biology, raising fears about the potential for creating biological weapons or other harmful applications. These are not simple questions, and they invite us to reflect deeply on our responsibilities as we continue to push the boundaries of biological innovation. Let us pause for a moment to consider the ethical landscape of human genome editing, a particularly sensitive area within genetic engineering. Technologies like CRISPR have made it significantly easier to edit the human genome, presenting both medical and non-medical applications, such as gene therapy. A major ethical issue at the forefront is the possibility that these technologies might be used to make permanent alterations in human genes that are capable of being passed on to future generations, to children yet to be born. In Canada, editing human genes for inheritable changes is explicitly restricted. The Assisted Human Reproduction Act of 2004 dictates that editing the human genome in a way that could be inherited is punishable by up to 10 years in prison. This means it is illegal for researchers to alter the human genome in any way that could affect eggs or sperm. However, gene editing of adult cells, which affects only the individual being treated and is not passed on to offspring, is generally permitted. This distinction reflects a careful balance, allowing therapeutic interventions for existing individuals while placing strict prohibitions on inheritable changes that could have unforeseen, long-term consequences for the human gene pool. It is a reflection of society's profound caution when dealing with the fundamental genetic blueprint of our species. The discussion around genetic engineering often invites comparisons with older forms of biological modification, highlighting how synthetic biology represents a qualitative shift. Genetic engineering itself is not entirely new. Humans have, in various forms, been influencing the traits of organisms for millennia. However, the advent of new technologies in this discipline allows us to play God in qualitatively different ways than ever before. Consider selective breeding, a practice as old as agriculture itself. Selective breeding is constrained by working within the natural timescales and genetic possibilities dictated by evolution. While artificial selection and traditional breeding techniques can achieve results faster than natural evolution, they are still limited to combining existing genetic variations within a species. Genetic engineering, however, partially frees us from this constraint, allowing us to modify genes directly, even introducing genes from entirely different species. Synthetic biology promises to free us from an even further constraint, the need for a natural template upon which future organisms must be based. It envisions a future where we can design and create life not merely by tinkering with or modifying existing forms, but by building entirely new biological systems from basic components, much like an engineer constructs a machine. This marks a profound shift in our relationship with the living world, moving from modifying to truly creating. The concept of playing God, or creating life from non-living, inorganic matter, is a central ethical concern that arises with synthetic biology. This field enables us to synthesize genetic material and construct organisms in a way that approaches generating life from scratch. The argument against synthetic biology often posits that its most distinctive role, that of creating novel biological systems, might well be a role traditionally assigned to a higher power or to nature itself. From this perspective, it is thought to require particular sensitivity to human limitations, suggesting that synthetic biology should therefore not be pursued by humans, as it oversteps our inherent boundaries. However, a counter-argument gently suggests a different perspective. It proposes that the modification, design, and creation of life could also be defended on the grounds that these endeavors promote human well-being, provided proper caution is taken. This view suggests that such activities are consistent with a natural or even a God-given desire for the improvement of the human condition. It argues that we are working with materials and intellectual resources that are ultimately provided by nature or a divine source, and therefore, our creative engagement with them is not an affront but rather an expression of our human potential. This delicate balance between awe and responsibility remains a central point of reflection in the ethical landscape of synthetic biology. Another profound ethical question raised by synthetic biology is whether its developments blur the distinction between organisms and machines. Synthetic biology often uses machine-like language extensively, referring to concepts such as genetic circuits or biobricks, which are modular biological parts. Under most traditional ethical frameworks, living things and machines are placed into firmly separate categories, each with distinct moral implications. Consider the example of creating bacterial biofactories by adding suitable modules to a minimal bacterial chassis. These engineered bacteria possess many characteristics of life, including homeostatic physiological mechanisms, a nucleic acid genome, protein-based structures, and the ability to reproduce. Yet, they also possess characteristics typically associated with machines, modular construction, based on rational design principles, and designed with specific applications in mind, often for a particular end goal. This dual nature challenges our conventional classifications and forces us to reconsider the very boundaries we draw between the natural and the artificial, between organisms and engineered devices. This blurring of lines forms a significant part of the ethical debate, prompting deeper reflection on how we value and define different forms of existence. This leads us to a deeper ethical concern: how much do we truly value life, and what constitutes its moral status? The concern is that with synthetic biology, we might inadvertently adopt a reductionist account of life, seeing it as nothing more than a collection of biochemical components. This perspective could undermine the special status traditionally afforded to living things and diminish the intrinsic value we ascribe to life. There's a significant difference in moral status between machines and living things. Machines, by definition, have no intrinsic value. They are valuable only insofar as they can be used to achieve an end goal. They possess no inherent interests and no rights. Organisms, on the other hand, are generally considered to possess moral status, with their own interests and, in some cases, rights. The problem arises if we wrongly perceive living things, especially those engineered through synthetic biology, as possessing less moral status, or conversely, if we mistakenly ascribe moral worth to what are essentially complex machines. This delicate philosophical distinction requires careful thought as our capacity to engineer life continues to advance. The question of whether moral status is conferred by life itself is complex and nuanced. It's often argued that moral status is not universally granted simply by being alive. For instance, most ethical frameworks do not typically ascribe moral status to all bacteria or plants in the same way they do to sentient animals or humans. This prompts us to ask, what, then, truly confers moral status? Is it sentience, the capacity to feel and perceive? Is it consciousness, the state of being aware of one's existence and surroundings? Or is it self-consciousness and rationality, the ability to reflect on oneself and reason? These are profound questions, and our conception of moral worth linked to such features can itself be problematic. For example, do all humans consistently meet our criteria for conscious and rational behavior at every stage of life, or in every state of being? This line of questioning reveals the deeply intricate nature of defining moral status and the challenges it poses when applied to newly engineered forms of life. The real concern, therefore, is not just about the definitions, but what determines moral status in practical terms. We generally agree that persons, with their complex inner lives and capacities, possess moral status, and machines, designed for specific functions, do not. However, there are many instances that fall into a gray area where our certainty is less firm. It is plausible that many simple synthetic entities, such as a minimal bacterial chassis, would possess no significant moral status. We can likely be confident in that. But what about more complex synthetic creations? For instance, we might be less confident about the moral status of a biocomputer constructed from synthetic human-like nerve cells. Such an entity would push the boundaries of what we consider machine and organism. This leads to a critical question: should we even attempt to create such a thing or being before we have thoroughly ascertained what moral status it would have? This underscores the urgent need to engage with the rich and thriving literature on the moral status of living things. We must use this body of knowledge to inform and develop our ethical framework for synthetic biology, ensuring that our advancements are guided by careful consideration and profound wisdom. Beyond the philosophical debates, there is a tangible concern about the misuse of knowledge generated by synthetic biology. The knowledge produced in this field, like many powerful scientific discoveries, might be intentionally misused for harmful purposes. This presents what is known as a dual-use dilemma, where the same scientific advancements can be applied in both beneficial and detrimental ways. Two groundbreaking studies illustrate this concern vividly. The first was the de novo synthesis of the poliovirus, a human pathogen, from its chemical components. This demonstrated the technical capacity to create dangerous viruses without relying on existing natural samples. The second study involved the accidental creation of a vaccine-resistant strain of the mousepox virus through traditional genetic engineering techniques. Both of these studies, while contributing to scientific understanding, raise deeply unsettling possibilities. They highlighted that states or even terrorist groups, perhaps without access to wild-type viruses, might acquire the ability to create human pathogens for use in biological weapons. It's a sobering thought, reinforcing the need for vigilance and ethical foresight in scientific dissemination. As a notable, and perhaps alarming, fun fact, the poliovirus genome, along with many other human pathogens, is actually in the public domain, meaning its genetic sequence is freely accessible, underscoring the inherent challenges in controlling knowledge itself. The dual-use dilemma is a central ethical challenge in synthetic biology, arising when scientific knowledge and technologies have the potential to be used for both good and harmful ways. The core of the dilemma lies in the risk of harmful use being sufficiently high that it becomes unclear whether such knowledge should even be pursued or widely disseminated. This is not a new problem in science. Historically, there have been precedents. Nuclear physicists in the first half of the last century grappled with this very dilemma when deciding whether to assist in the development of nuclear fission technology, knowing its potential for both energy generation and destructive weaponry. Similarly, research into behavioral genetics and racial differences in intelligence and character, as well as work on lie detection, DNA fingerprinting, biometrics, and traditional forms of genetic engineering, have all presented dual-use challenges. These historical examples remind us that the power of scientific discovery often comes with a profound responsibility to carefully consider its potential societal impact, both intended and unintended. The ethical controversies surrounding the dual-use dilemma in synthetic biology are profound. Our current attempts to synthesize designer pathogens would certainly be constrained by our limited understanding of how various complex factors combine to make a pathogen truly dangerous. However, a critical question arises: would engaging in high-tech synthetic techniques eventually become a cost-effective option for potential bioweapon proliferators, particularly for terrorist cells that may have limited resources? The answer is unsettling. It is plausible that in the future, as technological advances continue and costs dramatically reduce, the development of bioweapons using synthetic techniques could indeed fall within the capabilities of smaller and smaller groups, perhaps even lone biohackers. This prospect underscores the urgency of developing robust ethical guidelines and security measures to mitigate such risks, ensuring that scientific progress does not inadvertently empower malicious actors. The question of whether the threat posed by the misuse of knowledge from synthetic biology will ultimately be greater than that posed by nuclear technology is a serious one. While nuclear technology inherently requires bulky and expensive infrastructure, making it difficult for non-state actors to acquire, the technologies needed to produce bioweapons may, in contrast, become increasingly portable and affordable. This accessibility presents a unique and perhaps more pervasive risk. Furthermore, unlike advances in nuclear technology, which were often classified and kept confidential from their inception, the life sciences have a strong and long-standing tradition of openness. This means that much of the foundational background knowledge relevant to synthetic biology is already widely available in the public domain. This openness, while vital for scientific progress and collaboration, also means that the blueprints for potentially harmful biological constructs are more accessible. This inherent difference in transparency creates a unique challenge for controlling the misuse of synthetic biology knowledge, necessitating innovative approaches to biosecurity and responsible research practices. Considering these challenges, the discussion extends beyond merely the ethics of how scientific knowledge is produced. It encompasses the ethics of producing and, crucially, disseminating certain types of scientific knowledge. This requires us to develop thoughtful principles for determining when the creation or sharing of potentially dangerous knowledge is impermissible. It's a delicate balance, as restricting scientific communication can also stifle progress and transparency, which are cornerstones of scientific integrity. However, the potential for harm necessitates a careful re-evaluation of our traditional norms of open science. We must strive to create a framework that allows beneficial research to flourish while establishing clear boundaries and safeguards against the misuse of powerful biological tools and information. So, what can be done to address the dual-use dilemma and the potential misuse of synthetic biology knowledge? Certain downstream measures have already been implemented, aiming to manage the propagation of scientific knowledge without completely stifling its creation. For example, some scientific journals have adopted internal screening procedures. These procedures are designed to ensure that they do not publish material that presents an unacceptable risk of misuse, acting as a gatekeeper for potentially sensitive information. Furthermore, some funding bodies have begun to ask applicants to explicitly declare any risks that their proposed work might pose for misuse, encouraging researchers to think proactively about the broader implications of their studies. Governments, too, have become directly involved in influencing knowledge creation and dissemination. This includes implementing a range of personnel vetting procedures for students and researchers involved in sensitive areas of biology. These measures are intended to prevent dangerous knowledge from falling into the wrong hands, creating a multi-layered approach to biosecurity and responsible innovation in the field of synthetic biology. In considering how we decide on appropriate safeguards, there are three broad approaches to managing the risks associated with potentially dangerous scientific knowledge. The first is a laissez-faire, or leave-alone, approach. Under this model, scientists and their institutions are largely left to deal with, or even ignore, concerns about misuse as they see fit. This approach emphasizes scientific freedom but offers minimal external oversight. The second approach is a requirement for consideration. Here, scientists and their institutions are required to assess the risk of misuse posed by their work. However, they are still given the freedom to respond to those risks as they deem appropriate, without external mandates on specific actions. Finally, the third approach is a requirement for independent consideration. This is the most stringent, stipulating that certain projects or findings must be reviewed by an independent body before they can be funded, undertaken, or published. Each approach balances scientific autonomy with public safety in different ways, reflecting the ongoing debate about the most effective way to govern rapidly advancing technologies. The role of an ethics of knowledge is to help inform decisions between these alternative strategies, particularly when balancing the benefits of scientific progress against potential harms. This involves asking critical questions. Should it be required that there be absolutely no serious risk of harm, a precautionary test, before knowledge is generated or disseminated? Or should decisions be based on whether the expected benefits of the research clearly outweigh the expected harms, a harm-benefit test? Another consideration is whether there should be no clear and immediate risk of misuse, a proactionary test, before proceeding. Consider an example: if humans are naturally prone to overstate the risks or understate the benefits of new knowledge, then applying an overly precautionary or even a strict harm-benefit test might excessively stifle valuable scientific progress. Conversely, in many cases, attempting to influence the creation or dissemination of knowledge in synthetic biology might promote one value, such as security, at the expense of other important values, like economic growth, scientific freedom, or the intrinsic value of knowledge itself. Navigating these trade-offs requires a deep and thoughtful ethical framework that considers all facets of human endeavor and societal well-being. History offers notable examples of how the dissemination of certain information can have profound and unexpected consequences, illustrating the ethics of knowledge in action. In 1979, for instance, the Progressive magazine published the rough mechanism of the hydrogen bomb, complete with technical diagrams. This act exposed one of the best-kept secrets in scientific history. At the time of publication, only 4 governments were thought to possess this knowledge. The magazine argued that nuclear secrecy was fueling the Cold War by allowing nuclear policy to be determined by a security elite without proper public scrutiny. They contended that the potential cost of publishing the mechanism, even if it aided in acquiring H-bombs, was outweighed by the benefits of undermining that secrecy and promoting transparency. In a more recent and biologically focused event in 2006, a Guardian journalist successfully ordered a segment of the smallpox virus genome and had it delivered to his home address. This stunt highlighted a significant vulnerability: the ease with which potentially dangerous genetic sequences could be acquired. This incident prompted calls for better regulation of DNA sales and the control of benchtop DNA synthesizers, which could prevent the widespread distribution of such sequences. At a state level, strong enforcement of biological and chemical weapons conventions could also play a crucial role in mitigating the risks of misuse. These events underscore the continuous tension between the free flow of scientific information and the imperative to protect public safety. In conclusion, the bioethics of synthetic biology can be broadly understood through 3 major questions, among other critical concerns. First, does synthetic biology, in its capacity to design and create novel life forms, constitute an affront to what might be considered God-given or natural capabilities of humans? Are we, as a species, overstepping our inherent limitations by delving into the fundamental redesign of life? This question delves into our deepest philosophical and spiritual understandings of our place in the natural world. Second, does synthetic biology inherently undermine the moral distinction we traditionally draw between living things and machines? Are we, through our engineering approach to biology, unintentionally diminishing the moral worth of living things, or perhaps overestimating the moral value of what are essentially sophisticated biological machines? This line of inquiry challenges our ethical categories and how we assign value to different forms of existence. Third, does synthetic biology, by generating powerful new knowledge, make the misuse of such knowledge especially dangerous? Can we ethically control the deliberate misuse of this information, or effectively undermine the pernicious dissemination of potentially harmful knowledge? This addresses the practical and societal risks, particularly the dual-use dilemma. Fortunately, we are not navigating these questions without guidance. We can leverage existing literature, draw lessons from historical precedents, examine contemporary examples, and analyze outcomes to better understand these complex problems. This allows us to formulate a robust ethical framework to deal with them. While some of these concerns have been, and continue to be, addressed through ongoing dialogue and policy development, these fundamental questions persist and evolve as synthetic biology itself advances. This ongoing reflection is not a barrier to progress, but a necessary companion to truly responsible innovation. Now, let us gently transition into the more practical, yet still calmly presented, aspects of how genetic engineering and synthetic biology are actually done. We'll touch upon the building blocks, the tools, and the methods that allow scientists to interact with the very code of life, always keeping in mind the vast potential and the quiet responsibility that accompanies such work. Our journey into genetic engineering begins with understanding its fundamental aim: to work with the flow of genetic information within living systems. Molecular biology, the broader field that underpins genetic engineering, studies how information moves from DNA to RNA, and then from RNA to protein. Think of DNA as the master blueprint, RNA as the working copy, and protein as the functional product. This flow involves several key processes: DNA replication, where the blueprint is copied, DNA transcription, where the DNA instructions are copied into mRNA, and mRNA translation, where these mRNA instructions are used to build proteins. Genetic engineering, at its core, involves manipulating this flow. A central technique is molecular cloning, which refers to experimental methods used to create recombinant DNA molecules. Recombinant simply means DNA that has been combined from different sources. Essentially, molecular cloning is a strategy to replicate this natural flow of genetic information, but on the lab bench, allowing scientists to isolate, copy, and modify specific genes. This foundational understanding allows us to precisely target and alter the genetic instructions, opening up possibilities that range from understanding basic biological functions to creating new therapies. The traditional molecular cloning workflow can be imagined as a series of precise, gentle steps, much like carefully assembling a delicate model. It begins with the need to cut both a plasmid, which is a small, circular piece of DNA often found in bacteria, and the gene fragments that we want to insert. These cuts are made using special molecular scissors called restriction enzymes. These enzymes recognize specific sequences in the DNA and cleave it, creating ends that can then be joined. Once the plasmid and the gene fragment are cut, they can be joined together in a process called ligation. This is like using a molecular glue to connect the DNA pieces. The result is a recombinant plasmid, containing the desired gene fragment. This recombinant plasmid is then introduced into a bacterial cell in a process called transformation. Once inside, the bacterial cell can replicate the plasmid, making many copies of the new, engineered DNA. This entire process allows scientists to incorporate new genetic information into cells, essentially giving them new instructions or capabilities. After performing molecular cloning, it is crucial to verify that the cloning process was successful and that the desired gene has indeed been incorporated into the plasmid and taken up by the bacterial cells. This is achieved through several verification steps. First, the bacterial cells that have taken up the recombinant plasmid are grown on selective media. This media often contains an antibiotic, and because recombinant plasmids typically carry an antibiotic-resistant gene, only the bacteria that have successfully taken up the plasmid will survive and form colonies. This provides a clear selection mechanism. Once colonies are grown, the recombinant plasmid can be extracted and further analyzed. One common technique is restriction mapping. This involves cutting the plasmid with restriction enzymes and then running the resulting DNA fragments on a gel electrophoresis. The pattern of DNA bands on the gel can help confirm the size and structure of the plasmid, indicating whether the gene insert is present and in the correct orientation. Finally, sequencing, a technique that determines the exact order of nucleotides in the DNA, provides the ultimate verification. Sequencing confirms that the inserted gene is indeed present and that its genetic code is accurate, ensuring the successful creation of the engineered DNA molecule. At the heart of molecular cloning and many genetic engineering techniques is the plasmid. But what exactly is a plasmid? Imagine a bacterial cell. Most bacteria contain a single, large, circular chromosome, which holds the majority of the organism's genes, essential for its survival. Plasmids are much smaller, often circular, double-stranded DNA molecules that exist separately from this main chromosome. They are like tiny, independent genetic minichromosomes within the cell. Plasmids have several distinctive features. They replicate independently from the main chromosome, often using the same enzymes as the host cell. They are typically much smaller than chromosomes, ranging from 1 kilobase pair to over 1 megabase pair, usually less than 5% of the size of the main chromosome. Importantly, plasmids are usually not essential to the cell's basic survival under normal conditions, meaning the cell can live without them. However, they often carry genes that provide advantageous traits, such as antibiotic resistance. Furthermore, each bacterial cell can contain multiple copies of the same type of plasmid, or even different types of plasmids, making them incredibly useful vehicles for carrying and amplifying engineered genes. A recombinant plasmid, the engineered DNA molecule used in genetic engineering, contains several key elements, each with a specific function. One crucial element is the origin of replication, or WORI. This is a specific DNA sequence that directs the initiation of plasmid replication by recruiting the cell's transcription machinery. Essentially, the WORI controls how many copies of the plasmid are made within the host cell and determines the host range, meaning which types of cells the plasmid can successfully replicate in. It's important to note that plasmids with the same WORI are often incompatible, meaning they cannot stably coexist in the same bacteria because they compete for the same replication machinery, leading to an unstable and unpredictable environment. Different WORIs produce varying numbers of plasmid copies, from just a few to several hundred, so the best choice depends on the specific needs of the experiment. Another essential feature of a recombinant plasmid is a gene that provides an advantageous trait for survival, most commonly an antibiotic resistance gene. This allows scientists to select for cells that have successfully taken up the plasmid, as only those cells will survive in the presence of the antibiotic. Cloning sites are specific sequences within the plasmid that can be cut by restriction enzymes, allowing new DNA fragments to be inserted. A promoter is a sequence of 100 to 1000 base pairs found upstream of the target gene. It controls the binding of RNA polymerase and other transcription factors, initiating the transcription of the gene. The type and strength of the promoter are critical, as they dictate the level of gene expression. Some promoters are a constitutive, always active, while others are inducible, meaning they can be turned on or off. Lastly, the insert itself, containing restriction sites, a ribosome binding site, and primer binding sites, is crucial. Ribosome binding sites, like Schindal-Garneau sequences in bacteria, are short sequences 6 to 10 nucleotides upstream of the start codon that recruit ribosomes for translation. Primer binding sites are initiation points for PCR amplification or DNA sequencing, used to verify the insert sequence. Each of these elements is carefully chosen and assembled to create a functional genetic tool. One of the most foundational techniques in molecular biology and genetic engineering is the polymerase chain reaction, or PCR. The first objective of genetic engineering is often to isolate copies of specific genes in a pure, amplified form, and PCR is the workhorse for this task. DNA amplification, through PCR, is the process of generating multiple copies of a target DNA or RNA molecule, typically performed in vitro, meaning outside their normal biological context, in a test tube in the lab. This allows scientists to create millions or even billions of copies from a single starting molecule. Unlike the continuous replication that occurs within a living cell, DNA amplification via PCR requires a specific temperature cycle. It is not an isothermal process, meaning the temperature changes precisely throughout different stages. While PCR is the most common method, other isothermal DNA amplification techniques do exist, such as rolling circle amplification, loop-mediated amplification, and strand displacement amplification. These methods offer alternatives for situations where a constant temperature is preferred. However, for sheer power and versatility in amplifying specific DNA sequences, PCR remains an indispensable tool in the genetic engineer's arsenal. The polymerase chain reaction, or PCR, operates through a precise, cyclical process involving three main temperature-dependent steps. First, there's denaturation, typically occurring at around 95 degrees Celsius for about 1 minute. During this step, the high heat causes the double-stranded DNA template to separate into two single strands, as the hydrogen bonds holding them together break down. Next is annealing, where the temperature is lowered to around 55 degrees Celsius for about 45 seconds. In this crucial step, short, synthetic DNA sequences called primers bind, or anneal, to complementary regions on each of the separated single DNA strands. This is the process of allowing two sequences of DNA to form hydrogen bonds. The precise annealing temperature is critical for the primers to bind specifically to their target sequences without attaching elsewhere. Finally, elongation or extension occurs at approximately 72 degrees Celsius for 1 to 2 minutes. This temperature is optimal for the Taq polymerase, a heat-stable DNA polymerase enzyme isolated from a bacterium found in hot springs. The Taq polymerase binds to the annealed primers and begins adding new nucleotides, complementary to the template DNA strand, extending the primer to synthesize a new, double-stranded DNA molecule. This entire cycle of denaturation, annealing, and elongation can be repeated 24 to 30 times. With each round of amplification, the amount of target DNA effectively doubles, leading to an exponential increase in the specific DNA sequence, yielding millions to billions of copies from a tiny starting sample. Designing a successful PCR reaction involves asking several important questions and considering specific reagents. First, we need to design PCR primers, which are short, specific DNA sequences that define the start and end of the DNA region to be amplified. Then, we determine the number of cycles to run, typically 24 to 30, to achieve sufficient amplification. We also need to decide how long each step, denaturation, annealing, and extension, should run, as well as the precise temperatures for each step. The concentration of each reagent is critical for an efficient reaction. The reagents for PCR include template DNA. This is the DNA sample containing the target sequences we wish to amplify. Sequence-specific DNA primers. These are typically 18 to 24 bases long and flank the target sequence, with both a forward and a reverse primer. In PCR, unlike cellular replication, these primers are made of DNA. Taq polymerase. This is a DNA polymerase enzyme from Thermus aquaticus, a bacterium found in hot springs, making it highly resistant to the high temperatures of the denaturation step. It adds new nucleotides to the growing DNA strand. Deoxynucleotide triphosphates, DNTPs. These are the building blocks of DNA, DATP, DCTP, DGTP, and DTTP, which the Taq polymerase incorporates into the new DNA strands. PCR buffer. This solution maintains the optimal chemical environment, including pH and salt concentration, for the Taq polymerase to function efficiently. To ensure the reliability of a PCR reaction, negative controls are essential. A no template control, or NTC, omits any DNA template from the reaction. This serves as a general control for extraneous nucleic acid contamination, helping to detect if any unwanted DNA is amplifying from reagents or the environment. If a band appears in the NTC lane after PCR, it indicates contamination, and the results of the experimental samples cannot be trusted. Another important control is a no amplification control, or NAC. This control omits the DNA polymerase enzyme from the PCR reaction. The purpose of the NAC is to control for any background signal that might not be a function of the actual PCR amplification process itself. If there is any signal in the NAC, it suggests that something other than the intended enzymatic amplification is contributing to the observed result. These negative controls are vital safeguards, helping researchers to confidently interpret their PCR results and ensure the specificity and purity of their DNA amplification. The design of primers is a critical step in PCR, influencing the success and specificity of the amplification. Primers are short, single strands of RNA or DNA, typically about 18 to 24 bases in length. An important distinction between PCR and cellular replication is that primers in PCR are made of DNA, not RNA. These primers serve as the starting point for DNA synthesis, as DNA polymerases can only add new nucleotides to an existing strand of DNA. Researchers custom design these forward and reverse primers to be complementary to the beginning and end of their target DNA sequence. This ensures that both complementary strands of the target DNA are amplified simultaneously during the PCR cycles. Careful primer design is therefore fundamental to isolating and amplifying the specific genetic information of interest. The main property of primers is that they must correspond to sequences on the DNA template, though they don't always need to be perfectly complementary throughout their entire length. Several key properties are considered during primer design. Ideally, primers should be between 18 to 24 bases long. Their melting temperature, or TM, which is the temperature at which half of the DNA strands are denatured, should ideally be between 50 and 60 degrees Celsius. A good primer typically has a 40 to 60% GC content, as guanine and cytosine bases form stronger bonds than adenine and thymine. It's also beneficial for primers to start and end with 1 to 2 GC pairs, as strong binding at the ends, especially the 3' end, is important for stable attachment. Furthermore, primer pairs should have melting temperatures within 5 degrees Celsius of each other to ensure both anneal efficiently under the same reaction conditions. To avoid unwanted secondary structures, primer pairs should not have complementary regions that could cause them to bind to each other, forming primer dimers, or to self-anneal into hairpin loops. Finally, it's generally advisable to avoid long stretches of di nucleotides, like repeated AT or GC sequences, which can also lead to non-specific binding and reduce PCR efficiency. The length of the primer is indeed important. The more nucleotides you add, generally the more specific the primer becomes in binding to its target sequence. However, if primers are too long, they can result in a slower hybridizing rate, making the annealing step less efficient. Conversely, if you add too few nucleotides, making the primer too short, it may produce inaccurate and non-specific DNA amplification products, binding to unintended regions of the genome. Therefore, a length of 18 to 24 bases is generally considered ideal, striking a balance between specificity and efficient annealing to the template DNA, forming a stable, double-stranded DNA polymerase binding site. This careful balance ensures that the PCR reaction targets only the desired genetic sequence, maximizing accuracy and yield. The 3-foot end of a primer is absolutely critical for the success of a PCR reaction. Specifically, the last 5 to 6 nucleotides at this end play a crucial role. This is because DNA polymerase, the enzyme responsible for synthesizing new DNA strands, can only add new nucleotides to an existing free 3-foot hydroxyl group on the primer. Therefore, when designing primers, one must start by carefully considering the 3-foot end of the template DNA. Perfect base pairing between the 3-foot end of the primer and the target DNA template is essential for the polymerase to initiate synthesis accurately. Additionally, including guanine and cytosine bases in this crucial 3-foot section will increase binding to the template, as GC pairs form 3 hydrogen bonds, making the attachment more stable and robust. This precise interaction at the 3-foot end is fundamental to the specificity and efficiency of the entire PCR process. The melting temperature, or TM, of a primer is determined by the specific type of bases it contains. Primers with a higher number of guanine and cytosine bases will have a higher TM. This is because GC base pairs form 3 hydrogen bonds, which are stronger and require more energy to break compared to the 2 hydrogen bonds formed by adenine-thymine base pairs. Keeping the GC content of primers within the range of 40% to 60% is generally recommended as it optimizes amplification efficiency. Additionally, to prevent non-specific binding and the formation of unwanted secondary structures, it's important to try and avoid what are called GC clamps. These are sequences where there are more than 3 or 4 GS or CS consecutively at the 3rd end of the primer. Such clamps can lead to overly strong and non-specific binding, hindering the precise action of the polymerase. When determining the optimal melting temperature, or TM, for primers, generally, it should fall between 50 and 60 degrees Celsius. This range is crucial for efficient annealing, allowing the primers to bind specifically to their target sequences without attaching elsewhere. A TM higher than 65 degrees Celsius can sometimes lead to what's known as secondary annealing, where primers bind to unintended regions due to excessively strong interactions. A simple formula can be used to calculate TM. TM equals 2 degrees Celsius multiplied by the number of adenine and thymine bases, plus 4 degrees Celsius multiplied by the number of guanine and cytosine bases. For the annealing temperature, or TAR, of primers, it should ideally be within 5 degrees Celsius of each other for a primer pair. If the TM values of your primer pair vary by more than 2 to 5 degrees Celsius, the primer with the higher TM might suffer from a mispriming and at the lower annealing temperature, binding to non-specific sites. Conversely, the primer with the lower TM might not be functional at a very high annealing temperature, failing to bind at all. To calculate the TAR, a common rule of thumb is to subtract 5 degrees Celsius from the TM. It's important to remember that these calculations provide an educated guess and often require experimental fine-tuning in the lab. It's important to note that calculating the annealing temperature, or TAR, is not solely dependent on the composition of C, A, T, and G bases. Other factors, such as the pH of the reaction buffer and the concentration of salts present, also significantly influence the optimal temperature. Many companies that specialize in producing and selling primers offer their own TAR calculators. These calculators attempt to mimic some of the real-world conditions that will be present in your reaction tube, providing a more accurate estimate. However, it's crucial to keep in mind that these numbers are always at best an educated guess and serve as a starting point for your experiments. Typically, when setting up PCR samples, it's good practice to test more than one reaction condition. By varying the annealing temperature or other parameters slightly, you can pinpoint the optimal reaction conditions that yield the desired specific product and minimize unwanted amplification. This empirical testing ensures the most successful and reliable PCR results. To ensure the success of any PCR reaction, following some good rules can significantly improve outcomes. First, always strive to understand what you are doing and why. Knowing the purpose of each step and each reagent helps in troubleshooting and making informed decisions. Second, always double-check that your primers are correct, especially with respect to their complementarity to the target DNA sequence, to avoid non-specific binding. Third, utilize available computer programs to help determine your optimal annealing temperature, as these tools can provide valuable initial estimates. Fourth, don't be afraid to test more than one annealing temperature condition in preliminary experiments to find the most efficient and specific one. Fifth, use a checklist when adding reagents to your reaction tubes. This simple practice can prevent costly mistakes and ensure all necessary components are included in the correct concentrations. Finally, always double, or even triple, check your reagents and ensure the proper concentrations for each component you need to add for the reaction. These meticulous practices are key to achieving reliable and reproducible results in PCR. When it comes to obtaining primers for your genetic engineering projects, several specialized companies and software tools are available to assist researchers. Companies like Integrated DNA Technologies, IDT, are major suppliers of custom synthesized DNA and RNA oligonucleotides, including the primers essential for PCR. These companies offer services for designing and synthesizing high-quality, sequence-specific primers to meet the precise needs of any experiment. Beyond commercial suppliers, sophisticated software tools are invaluable for primer design and experiment simulation. Benchling, for example, is a unified research and development platform. It allows users to simulate and design experiments, offering automated primer design, tools for restriction digests, and PCR simulation capabilities. This integrated platform helps researchers plan and visualize their experiments before physically conducting them. Another powerful tool is NCBI Blast, or basic local alignment search tool. Within NCBI, PrimerBlast is a web application specifically designed to help users create target-specific primers. It offers flexibility to accommodate various primer design criteria, allowing users to design new primers or check the specificity of pre-existing ones against a vast database of genetic sequences. These resources are indispensable for modern genetic engineers. The applications of PCR are incredibly broad and have revolutionized many different laboratory procedures. Once DNA has been amplified through PCR, the numerous copies can be used for a wide array of scientific and practical purposes. In genetic research, PCR has been fundamental. For example, mapping techniques used in the Human Genome Project relied heavily on PCR to amplify specific regions of DNA for analysis. PCR is also central to DNA fingerprinting, a technique used for identification in forensic science and paternity testing by analyzing unique genetic markers. It is crucial for the detection of bacteria or viruses, allowing for highly sensitive and rapid diagnosis of infections, such as HIV, by amplifying even tiny amounts of pathogen DNA or RNA present in a sample. Furthermore, PCR is vital in the diagnosis of genetic disorders, enabling the identification of specific mutations associated with various inherited diseases. These are just a few examples. The versatility of PCR means it has many more applications across biology, medicine, and biotechnology, making it an indispensable tool for understanding and manipulating genetic information. Adding restriction sites via PCR is a clever technique used to prepare DNA fragments for molecular cloning. Restriction sites, also known as restriction recognition sites, are specific sequences of nucleotides, typically 4 to 8 base pairs long, located on a DNA molecule. These sites are recognized and cut by restriction enzymes, which act as molecular scissors, cleaving double-stranded DNA at these precise locations. The goal of adding these sites is to create compatible ends that can be easily joined with a linearized plasmid, which is essentially a circular plasmid that has been cut open. When conducting an overlap extension PCR, primers are designed with a small overhang that includes the desired restriction site. After amplification, the resulting gene fragment will have these new restriction sites at its ends. This allows the amplified gene fragment to be cut with the corresponding restriction enzymes, creating sticky ends or blunt ends that are compatible with the prepared plasmid backbone. The plasmid backbone, often a circular piece of DNA, will also have been cut with the same restriction enzymes, creating matching ends. This ensures that the gene fragment can be ligated, or glued, into the plasmid in the correct orientation, enabling its subsequent use in genetic engineering. Gel electrophoresis is a fundamental molecular biology technique used to separate and, sometimes, purify DNA, RNA, or proteins based on their physical properties. This separation is primarily governed by 3 factors: the molecule's charge, its size, and its conformation or shape. DNA and RNA molecules are inherently negatively charged due to their phosphate backbone, which means they will naturally travel towards the positive electrode in an electric field. The process involves placing the samples in wells within a porous matrix gel. As an electric current is applied, the negatively charged DNA and RNA molecules migrate through the gel. Smaller molecules can navigate the pores of the gel more easily and thus travel faster and further than larger molecules. This results in a separation of DNA fragments by size, with lighter fragments moving faster and heavier fragments moving slower, forming distinct bands. Common gels used for this purpose include agarose, a complex sugar derived from seaweed, and polyacrylamide. The electrical resistance of agarose gel is similar to that of the surrounding buffer, which is why the same buffer solution is typically used for both making the gel and running the electrophoresis. This ensures a consistent electrical environment for separation. When visualizing the results of gel electrophoresis, we stain the DNA with a fluorescent dye that binds to the DNA. Once stained, the gel is placed on an ultraviolet transilluminator, which causes the stained DNA bands to glow brightly, making them visible. One common dye is ethidium bromide, or EtBr. This is a flat molecule that intercalates, or fits between, the adjacent base pairs in the DNA double helix. When EtBr is intercalated into DNA, its fluorescence significantly increases compared to when it's free in an aqueous solution. It can detect as little as 1 to 5 nanograms of DNA. However, EDBR is a mutagen, so safer alternatives are widely used today, such as SYBR Safe or GelRed. These dyes function similarly, binding to DNA and fluorescing under UV light, allowing researchers to accurately assess the size and quantity of their DNA fragments. This visualization step is crucial for confirming the success of PCR, restriction digests, or other DNA manipulation experiments. For the analysis of extracted DNA, a powerful tool is the DNA ladder or DNA marker. This is a solution of double-stranded DNA fragments of known molecular weights and specific numbers of base pairs. These markers are standardized, often consisting of DNA that has been cut from a plasmid or bacteriophage, providing a reference for determining the size of unknown DNA fragments in experimental samples. When run alongside your samples on a gel, the ladder creates a series of visible bands, each representing a specific DNA size. To perform analysis, once the DNA is separated on the gel and visualized, researchers often excise or carefully cut out the DNA band of interest from the gel. This purified DNA can then be used for further studies. The intensity of the bands on the gel can also give an approximate indication of the amount of DNA present. If some bands appear thicker, it generally means there is a higher concentration of DNA fragments of that particular size. This combination of size determination and approximate quantification makes gel electrophoresis an indispensable technique for molecular biologists. To help visualize the progress of DNA during gel electrophoresis, tracking dyes, also known as loading dyes, are added to the DNA samples. These dyes move towards the positive electrode at a predictable rate, independent of the agarose gel concentration, typically within a range of 0.5% to 1.4%. Two common tracking dyes are bromophenol blue, which moves at approximately the same speed as DNA fragments consisting of 300 base pairs, and Zion cyanol, which travels at a similar rate to DNA fragments around 4,000 base pairs long. It's important to understand that these tracking dyes do not directly influence the visibility of the DNA fragments themselves, they simply serve as visual indicators. By comparing the position of a DNA fragment in the gel to the position of these dyes, technicians can see the approximate number of nucleotides the DNA fragment contains without having to wait for the DNA to be fully separated and stained. This allows researchers to monitor the progress of the electrophoresis and stop the run at the optimal time, ensuring efficient separation. The composition of the gel, particularly its porosity, significantly impacts how DNA fragments migrate during electrophoresis. Porosity is a measure of how many pores are present within a given agarose gel. A higher gel concentration results in decreased porosity, meaning fewer and smaller pores. Consequently, smaller pore sizes create more resistance, making it harder for DNA molecules to migrate through the gel. This also means that in a more concentrated agarose gel, the largest pores that exist are likely smaller than those found in a less concentrated gel. The pore size distribution, which is the range of sizes of pores within the gel, is critical. More pores and larger pores generally facilitate the migration of larger DNA molecules more effectively through the gel. Conversely, fewer and smaller pores lead to greater distinction and better separation between smaller DNA molecules. This fine-tuning of gel concentration allows researchers to optimize the separation for different sizes of DNA fragments, ensuring clear and accurate results. There are two main types of gels commonly used for DNA electrophoresis: agarose and polyacrylamide gels, each suited for different applications. Agarose, a complex sugar derived from seaweed, is widely used for larger DNA fragments. Agarose gels are typically run horizontally. A key characteristic of agarose gels is that the higher the agarose concentration, the smaller the pore sizes within the gel. This means that DNA molecules will migrate a shorter distance. Agarose gels are generally used at concentrations between 0.5% and 2%, and their pore sizes, which are not uniform, range from approximately 50 to 200 nanometers. This makes them ideal for separating DNA fragments ranging from 50 to 20,000 base pairs in size, making them particularly good for analyzing large DNA fragments. A significant advantage of agarose is that it is non-toxic, making it safer to handle in the lab. Polyacrylamide gels, on the other hand, are prepared by chemically cross-linking acrylamide and bis acrylamide. The pore sizes in polyacrylamide gels are determined by the ratio of acrylamide to bis acrylamide and are notably more uniform than in agarose gels. These gels are usually made at a concentration of 6% to 15% and offer very high resolving power, making them excellent for separating small DNA fragments, typically from 5 to 500 base pairs. This high resolution also makes them suitable for analyzing single-stranded DNA and proteins. However, it is important to note that acrylamide is a potent neurotoxin in its monomeric form, requiring careful handling precautions during gel preparation. Each gel type serves a specific purpose in the diverse world of molecular biology techniques. The migration rate of DNA through a gel is influenced by several factors. The molecular size of the DNA is paramount. The rate of migration is inversely proportional to the logarithm of the number of base pairs, meaning smaller molecules move faster. The conformation, or three-dimensional shape, of the DNA also matters. Supercoiled DNA, which is tightly wound, migrates differently than linear DNA or relaxed circular DNA, offering useful controls. The presence of ethidium bromide, or ETBr, in the gel or buffer can affect migration. ETBr intercalates into DNA, which decreases its negative charge and increases its stiffness and length, thereby retarding the migration rate by about 15%. The applied voltage is another factor. For maximum resolution of DNA fragments larger than 2 kilobase pairs, a lower voltage of 1 to 5 volts per centimeter is often used. The type of agarose also plays a role, as different types offer varying separation ranges. Finally, the electrophoresis buffer, which conducts electricity, is crucial. Its composition and ionic strength affect migration. If there are no ions, DNA won't move. If there are too many ions, it can generate too much heat. Common buffers like TAE, triacetate EDTA, offer faster migration and higher resolving power for larger DNA, while TBE, trisborate EDTA, is another widely used option. After separating DNA fragments by size using gel electrophoresis, restriction mapping allows us to locate specific cutting sites within a DNA sequence. A restriction map is essentially a molecular blueprint, showing the known restriction sites within a DNA molecule. These maps are invaluable in molecular biology, serving as a reference for engineering plasmids or other DNA strands. One way to create a restriction map is to sequence the entire DNA molecule and then use a computer program to identify all known restriction enzyme recognition sites. The experimental procedure involves digesting purified plasmid DNA with various restriction enzymes, both individually and in combinations. The resulting DNA fragments are then run on a gel, and their lengths are determined by comparing them to a DNA ladder. By analyzing the sizes of the cleaved fragments from these single and double digests, the locations of the restriction sites can be inferred, allowing researchers to construct an accurate order of these sites along the entire DNA sequence. This process is like solving a puzzle, where each cut provides a clue to the internal arrangement of the DNA. Now, let us turn our attention to restriction enzymes themselves, the molecular scissors of genetic engineering. These enzymes are remarkable biological tools that cut double-stranded DNA at specific locations, known as restriction sites. They are found naturally in many prokaryotes, such as bacteria, where they serve as a defense mechanism against invading bacteriophages, which are viruses that infect bacteria. An interesting question arises: why isn't the host bacteria's own DNA affected by its restriction enzymes? The answer lies in another clever biological mechanism. The host DNA is protected by special enzymes called methylases. Methylases add methyl groups to the adenine and cytosine bases within the host's DNA at the specific recognition sites. This chemical modification acts like a protective tag, preventing the restriction enzymes from cutting the host's own genetic material while still allowing them to target and destroy foreign, unmethylated viral DNA. This elegant system highlights the precision and sophistication inherent in microbial defense mechanisms, which genetic engineers have learned to harness for their own purposes. Restriction enzymes operate through a precise, shape-to-shape matching mechanism. When a restriction enzyme encounters a DNA sequence that matches its specific recognition site, it gently wraps around the DNA molecule. This interaction causes a break in both strands of the DNA molecule. Different restriction enzymes recognize different specific sequences, typically ranging from 4 to 8 nucleotides in length. This specificity is why they are often named after the genus and species of the organism from which they were discovered. For example, E. coli was the first restriction enzyme isolated from Escherichia coli strain RY13. After the DNA is cut, the pieces need to be rejoined. This is where DNA ligase comes into play. DNA ligase is an enzyme that catalyzes the formation of a phosphodiester bond, which is essentially the chemical backbone of the DNA. For successful ligation, the DNA fragments must have been cut with the same restriction enzymes, ensuring that their ends are compatible. This precise cutting and rejoining ability allows scientists to cut out a gene from one source and insert it into another, forming recombinant DNA. When restriction enzymes cleave DNA at their recognition sites, they can produce different types of ends, which are crucial for subsequent ligation steps. One type is sticky ends, which are single-stranded overhangs. This occurs when the restriction enzyme cuts the double-stranded DNA in a staggered fashion, leaving a short, unpaired sequence of nucleotides on one strand. These sticky ends are highly desirable in cloning processes because they are complementary to each other, meaning they can easily form hydrogen bonds and sticks together. This makes sticky end ligation very efficient. Importantly, sticky end ligations are typically performed at lower temperatures, around 12 to 16 degrees Celsius, or even at room temperature, because these lower temperatures help to stabilize the hydrogen bonding between the complementary sticky ends. This precise and efficient joining is what makes sticky end ligation, with the help of DNA ligase, the glue that allows scientists to assemble new DNA molecules. In contrast to sticky ends, some restriction enzymes produce blunt ends. This occurs when the restriction enzyme cleaves the double-stranded DNA straight down the middle of the recognition sequence, leaving no overhanging bases on either strand. Both ends of the DNA fragment are perfectly flush. Blunt end ligation, while possible, is significantly less efficient than sticky end ligation, often being about 100 times slower. This is because there are no complementary overhangs to guide the joining process, making it harder for the DNA fragments to come together in the correct orientation. To improve the efficiency of blunt end ligation, scientists sometimes use a technique called A-tailing, where a single AID9 nucleotide is added to the 3-foot end of the DNA fragments, creating a short overhang. Alternatively, other single base overhangs can be added. Another challenge with blunt ends is self-ligation, where the ends of a single DNA fragment can ligate back onto themselves, especially if it's a vector without an insert. To prevent this, a process called dephosphorylation can be performed on the vector, removing phosphate groups from its ends and making it unable to self-ligate, thereby favoring the insertion of a desired DNA fragment. Each type of DNA end requires specific considerations to ensure successful assembly of genetic constructs. Ligation, the process of joining DNA fragments, is fundamental to creating recombinant DNA. DNA ligase is the enzyme responsible for this, essentially connecting two strands of DNA together. It forms an ester bond between the phosphate group of one DNA strand and the deoxyribose sugar of another, thereby sealing the backbone of the DNA molecule. In living cells, DNA ligase is an integral part of the cellular replication machinery, crucial for joining the short Okazaki fragment synthesized on the lagging strand during DNA replication. This highlights that DNA ligase is ubiquitous and absolutely essential for all forms of life. For cloning purposes, DNA ligase is often isolated from two main sources. One source is E. coli bacteria. This ligase uses energy gained by cleaving NAD, nicotinamide AID9 dinucleotide, but it has some limitations. It typically does not ligate blunt-ended DNA efficiently, and it cannot effectively join RNA to DNA. The other, more commonly used source, is bacteriophage T4. T4 DNA ligase is highly versatile. It can ligate the ends of DNA, oligonucleotides, short synthetic DNA or RNA molecules, RNA, and even RNA-DNA hybrids. Crucially, it can ligate both sticky ends and blunt ends, making it the preferred choice for many cloning applications. However, T4 DNA ligase requires ATP, adenosine triphosphate, as a cofactor to provide the energy needed for the ligation reaction. Each type of ligase has its specific strengths and requirements, allowing scientists to choose the best tool for their particular DNA joining task. The temperature at which ligation is performed is a critical factor influencing its efficiency. Ligation is generally easier and more efficient at lower temperatures. This is because at higher temperatures, all molecules, including DNA fragments and ligase enzymes, move faster. While this increased motion might seem beneficial, it actually makes it harder for the two DNA ends to collide and remain stably together long enough for the ligase to act. For sticky ends, lower temperatures are particularly advantageous because they help stabilize the hydrogen bonding between the complementary nucleotides of the overhangs, allowing them to stick together more securely. While DNA ligase itself has an optimal activity at around 25 degrees Celsius, the optimal temperature for physically bringing DNA ends together to form these stable complementary bonds is actually closer to 1 degree Celsius. Therefore, to strike a balance, ligation reactions are typically carried out at about 16 degrees Celsius for approximately 1 hour. However, to further increase the efficiency of bringing DNA ends together, especially for challenging ligations, the temperature can be lowered to 4 degrees Celsius for a longer incubation time, often overnight. Following ligation, it's common practice to heat and activate the ligase at 65 degrees Celsius for 10 minutes and then chill the reaction on ice before transforming it into competent cells. After performing a ligation reaction, it's important to check its success and efficiency. One common method is to mix a small portion of the ligation reaction with a loading dye that contains SDS, sodium dodecyl sulfate, and then run it on a standard agarose gel. SDS is an ionic detergent that disrupts non-covalent bonds in proteins, binding with high affinity. If the loading dye does not contain SDS, a smear of DNA might appear on the gel, making it difficult to judge the ligation result, as DNA-protein interactions could cause aggregation. However, with SDS, which eliminates these interactions, distinct bands should appear. Successfully ligated DNA, especially plasmids that have incorporated inserts, will typically appear as several high molecular weight bands on the gel, indicating that larger DNA molecules have been formed. By comparing these bands to a DNA ladder which contains fragments of known sizes, researchers can assess whether the desired ligated products are present and in what quantities. This visual check provides a quick and effective way to determine if this molecular glue has done its job and if the DNA fragments have been successfully joined together. Beyond traditional ligation, several advanced DNA assembly techniques have emerged, offering more flexible and efficient ways to construct complex genetic systems. Traditional molecular cloning, as we've discussed, relies heavily on restriction endonuclease and subsequent ligation, often leading to a scar or a small, non-coding sequence at the junction of the joined fragments. This approach is sequence-dependent, meaning specific restriction sites must be present, and it can limit the flexibility of design. The BioBrick synthetic biology approach, for example, aimed to standardize parts using specific restriction sites like EcoRI, SPI, and SPI. While this created an open-source registry of standard parts, the use of compatible restriction sites could sometimes destroy the recognition site itself, also creating a scar in the final construct. In contrast, newer DNA assembly techniques are often sequence-independent and based on overlaps between adjacent parts. Methods like Gibson assembly, CPEC, SLIC, and ligase cycling offer ways to join multiple DNA fragments in a single, scarless reaction. These techniques are particularly valuable for assembling many fragments into a complex biological device, overcoming some of the limitations of traditional restriction ligation cloning and enabling more ambitious genetic engineering projects. Gibson assembly is one such major workhorse of synthetic biology projects worldwide, developed by Daniel G. Gibson and his colleagues at the J. Craig Venter Institute. This powerful technique allows for the seamless assembly of multiple DNA fragments in a single, isothermal reaction, meaning it occurs at a constant temperature, typically 50 degrees Celsius for 15 to 60 minutes. It's particularly effective for assembling up to 6 fragments and is generally not ideal for very small fragments, those less than 100 base pairs. The method employs a mix of 3 enzymes, an exonuclease, a DNA polymerase, and a DNA ligase. First, an exonuclease enzyme chews back the ends of double-stranded DNA fragments, creating single-stranded overhangs that are complementary to adjacent fragments. Next, a DNA polymerase enzyme builds back the missing nucleotides in these single-stranded regions, extending the complementary overlaps. Finally, a DNA ligase enzyme stitches, or sticks, the two DNA pieces together by forming phosphodiester bonds, sealing the nicks in the DNA backbone. This elegant enzymatic process allows for the precise and scarless assembly of DNA fragments, making it a highly valuable tool for constructing complex genetic circuits. Golden Gate assembly represents another powerful DNA assembly technique, particularly useful for constructing highly complex systems involving 20 or more fragments. This method leveraged the unique properties of type IIS restriction enzymes. Unlike traditional restriction enzymes that cut within their recognition sequence, type IIS enzymes cleave DNA outside of their recognition sequence. This crucial feature allows Golden Gate assembly to create unique overhangs without leaving behind any scar sequences, ensuring a seamless and precise assembly of multiple DNA parts. The process often begins with designing DNA fragments that have specific 4 base pair overhangs at their ends. These overhangs are carefully chosen to be complementary to the adjacent fragments in the desired order. When a type IIS enzyme, a ligase, and the various fragments are combined in a single reaction, the enzyme simultaneously cuts the DNA and the ligase joins the appropriate fragments. The beauty of this system is that the type IIS enzyme's recognition site is located outside the cleavage site, so the recognition site itself is typically removed during the cutting process. This means that once the fragments are correctly ligated, the restriction sites are no longer present in the assembled construct, preventing further cutting and ensuring a stable, scarless final product. This elegant design makes Golden Gate assembly incredibly efficient for building intricate biological circuits with high fidelity. To effectively design fragments for DNA assembly, especially using techniques like Golden Gate, a careful planning process is followed. This often involves starting with a vector, which is the foundational plasmid into which new DNA will be inserted. This vector is designed with specific type IIS recognition sequences that are located away from the crucial genetic elements. The key is to engineer for BP overhangs on the fragments you want to assemble. The next step involves generating the individual DNA fragments, often through PCR amplification. Each fragment is designed to have specific 4 base pair overhangs at its ends. These overhangs are unique to each junction point and are complementary to the overhangs of the adjacent fragments in the desired assembly order. After PCR, these fragments are then subjected to a digestion step using the type IIS restriction enzyme. This enzyme cuts just outside the recognition site, revealing the designed 4 BP overhangs. Because these overhangs are unique and complementary to their neighbors, they will preferentially ligate together in the correct order, creating the final, seamless assembled DNA construct. This precise control over the ends of the DNA fragments is what allows for the modular and efficient construction of complex genetic systems. To summarize the various DNA assembly techniques, we can categorize them into 2 main types based on whether they are sequence-dependent or sequence-independent. Sequence-dependent methods, such as those utilizing restriction endonuclease, including the BioBrick and type IIS, like Golden Gate, approaches, rely on the presence of specific recognition sites within the DNA. These methods are precise but can sometimes lead to scar formation, where small, non-coding sequences are left at the junctions of joined DNA fragments. In contrast, sequence-independent methods are based on overlaps between adjacent DNA parts. These include techniques like Gibson assembly, CPEC, circular polymerase extension cloning, SLIC, sequence and ligation-independent cloning, and ligase cycling. A major advantage of these overlap-based methods is that they are scarless, meaning they produce seamless junctions without introducing any extra base pairs. They are also highly versatile for assembling multiple fragments simultaneously. While each method has its strengths and optimal applications, the common goal across all DNA assembly techniques is to efficiently and accurately construct complex genetic sequences from smaller, defined parts. DNA extraction is a foundational process in genetic engineering, serving several crucial purposes. Its primary objective is to obtain DNA from cells. Beyond simply isolating it, the process aims to purify DNA from various contaminants that are also present within cells, such as proteins, carbohydrates, lipids, and other types of nucleic acids like RNA. This purification step is essential because these contaminants can interfere with subsequent molecular biology applications. Once the DNA is extracted and purified, it can then be used for a wide range of further studies or applications in genetic engineering, providing a clean and usable sample of genetic material. This careful preparation ensures the reliability and accuracy of downstream experiments. The purified DNA obtained through extraction holds immense potential for various applications across different fields. In genetic engineering of plants and animals, DNA with desirable genetic traits can be extracted, purified, and then precisely transplanted into another organism's genome, leading to crops with improved resistance or animals with enhanced characteristics. For pharmaceutical products, recombinant genetics, which often relies on purified DNA, is used to produce vital medicines. Examples include the hepatitis B vaccine and human growth hormone. Insulin, a hormone crucial for diabetes management, is one of the most common pharmaceuticals created using DNA extraction and recombinant techniques. In medical diagnosis, DNA extracted from a patient is purified and analyzed to diagnose genetic conditions like cystic fibrosis or Huntington's disease, allowing for early detection and personalized treatment strategies. Finally, for identity verification, genetic fingerprinting is a powerful process that matches genetic material from an individual with other available genetic material. This has applications in forensic science, criminal investigations, and for determining biological relationships, such as in paternity testing. These diverse uses highlight the transformative impact of DNA extraction as a gateway to understanding, manipulating, and applying genetic information. The process of DNA extraction typically involves three main steps, each designed to progressively isolate and purify the genetic material. The first step is lysis. This crucial stage involves breaking open the cell, whether it's a bacterial cell, a plant cell, or an animal cell, to release its contents, including the DNA. This can be achieved through mechanical disruption, like homogenization or using a mortar and pestle, or through chemical lysis, using detergents like SDS to break down cell membranes, and buffering salts such as TriCl and EDTA to protect the DNA and inhibit enzymes that could degrade it. The second step is separation. Once the cells are lysed, the DNA must be separated from other unwanted cellular components. This involves processes like protein denaturation or precipitation, often using enzymes like proteinase K to degrade proteins, or techniques like phenol chloroform extraction, where DNA partitions into an aqueous phase while proteins go into an organic phase. Chemical methods, like using guanidine hydrochloride, or salting out with high concentrations of salts such as LiCl or NaCl at low pH, can also precipitate proteins, leaving DNA in solution. The goal is to remove as many contaminants as possible. The third and final step is purification. This involves further refining the DNA sample to remove any remaining unwanted material. This often includes RNA denaturation using enzymes like RNase to degrade RNA, followed by DNA precipitation using alcohol, such as ethanol or isopropanol. The DNA, now visible as a cloudy precipitate, can be collected and washed with 70% ethanol to remove residual salts. Finally, the purified DNA is resuspended in a buffer, typically TE buffer, TriHCl and EDTA, which protects it from degradation and prepares it for downstream applications. This meticulous three-step process ensures a high-quality, pure DNA sample. After the DNA has been successfully extracted, its quality and characteristics need to be analyzed before it can be used in further experiments. One primary method for analyzing extracted DNA is gel electrophoresis, which we discussed earlier. By running the extracted DNA on a gel, researchers can visually assess the quality of the DNA. If the DNA appears as distinct, high molecular weight bands without smearing, it indicates minimal DNA degradation, meaning the DNA molecules are largely intact. Conversely, if there is significant smearing or many small fragments, it suggests extensive DNA degradation. Gel electrophoresis also allows for the determination of the size of the isolated DNA fragments, which is crucial for confirming that the extraction process yielded DNA of the expected length. This visual and size-based analysis provides immediate feedback on the success of the DNA extraction. Another powerful method for analyzing extracted DNA is spectrophotometry. This technique allows for the quantitative determination of DNA concentration by measuring the absorbance of light at 260 nanometers. Nucleic acids, including DNA, absorb ultraviolet light most strongly at this wavelength. A standard conversion is that an absorbance reading of 1 at 260 nanometers corresponds to approximately 50 micrograms per milliliter of DNA. Spectrophotometry also allows for the determination of DNA purity by calculating the A260-Se280 ratio. This ratio compares the absorbance at 260 nanometers, DNA, to the absorbance at 280 nanometers, proteins, which absorb at this wavelength. A ratio of less than 1.6 typically indicates contamination, usually with protein. A ratio of 1.8 is considered indicative of pure DNA. If the ratio is greater than 2.0, it suggests contamination with RNA, which also absorbs UV light, though with a peak at 260 nanometers. This combined analysis of concentration and purity ensures that the extracted DNA is suitable for sensitive downstream applications, such as sequencing or cloning, where contaminants can interfere with enzymatic reactions and accurate results. Restriction enzyme mapping is a key technique for understanding the internal organization of a DNA molecule. As we've explored, a restriction map is essentially a blueprint that shows the locations of known restriction sites within a specific DNA sequence. These maps are fundamental reference tools in molecular biology, used to guide the engineering of plasmids and other DNA constructs. One way to create such a map is by sequencing the entire DNA molecule and then computationally identifying all recognition sites for various known restriction enzymes. However, often the mapping is done experimentally. The experimental procedure involves digesting purified plasmid DNA with individual restriction enzymes, and then with combinations of 2 or more enzymes. The resulting DNA fragments are then separated by size using gel electrophoresis. By analyzing the lengths of these fragments, researchers can infer the relative positions of the restriction sites. For example, if a circular plasmid of 20 kilobases is cut with one enzyme and yields a single 20 kb band, it means there is only one restriction site for that enzyme. If it's cut with another enzyme and yields 2 fragments, say 12 kb and 8 kb, it indicates 2 restriction sites. By systematically cutting and observing fragment sizes, the locations of the restriction sites can be accurately ordered in relation to the entire sequence, providing a detailed map of the DNA. Now, let us turn our attention to the fascinating world of synthetic biology, which focuses on building biological circuits. Synthetic biology aims to rationally construct complex biological systems from well-characterized components. This approach draws inspiration from engineering principles, where complex systems are built from standardized, predictable parts. Mathematical models and model-based computer-aided design tools play a crucial role in synthetic biology. They help researchers design and manufacture intricate systems composed of many interacting parts, allowing for prediction and optimization before physical construction. A model, in this context, is a representation of the essential aspects of a system. It presents knowledge of that system in a usable form for analysis or design. Essentially, it's a simplified description of reality, focusing on what's most relevant to a specific question. A model is always designed to answer a particular question. There is no single model that fits all purposes. The question being asked dictates the level of abstraction and the type of model that needs to be built. This allows scientists to focus on different aspects of a biological system, from the behavior of individual molecules to the interactions of entire populations of cells, depending on what they wish to understand or engineer. The process of building a mathematical model for protein expression in synthetic biology involves a systematic, step-by-step approach. First, you must define the molecular players and their interactions. This includes identifying all the relevant parts or molecules, such as transcription factors, DNA, mRNA, and proteins, as well as understanding how they interact with each other to make up your biological devices and the entire system. Second, you describe these molecules and interactions within a mathematical model. Molecular concentrations will typically become the variables of your model. The interactions between molecules, such as binding, transcription, translation, or degradation, will be described by mathematical functions within the model. We will cover some common reaction functions used to represent these biological processes. Third, you estimate biologically feasible parameter values for the model. One way to do this is to experimentally measure the parameters for each reaction directly in the lab. Alternatively, reverse engineering can be used, where model parameters are estimated by fitting the model's output to available experimental data, refining the model until it accurately reflects observed biological behavior. Finally, you analyze the dynamical behavior of the model. This involves testing how changes in specific parameters might affect the model's behavior, which helps to assess the model's robustness and predictability. You can also simulate knockout experiments, where certain molecules or interactions are removed from the system, and compare these simulated results with actual experimental data. This analysis step provides crucial feedback, allowing you to iterate and improve the previous 3 steps, continuously refining your model for greater accuracy and utility in designing biological circuits. Mathematical modeling in synthetic biology inherently involves a trade-off between accuracy and simplicity. Complex models, by their nature, aim to incorporate more parameters and interactions, thereby striving for greater accuracy in representing biological systems. However, a key challenge is that the precise values for all these parameters may not always be readily available through experimentation. Moreover, highly complex models can become harder to analyze and understand, making it difficult to extract clear insights or predict behavior. The goal of a good model is to strike a balance. It should accurately capture the fundamental aspects of the system while intelligently omitting irrelevant details that do not pertain to the specific question being asked. The modeling process must carefully consider the appropriate time and spatial scales for the system being studied, the type of experimental data that is available, and the most suitable types of simulation, analysis, and design tools to be used. Ultimately, a good model is characterized by 4 key traits: accuracy in describing current observations, predictability of system behavior in unobserved situations, reusability in similar cases, and parsimony, meaning it should be as simple as possible while still achieving its objectives. These characteristics ensure that models are both useful and manageable in the quest to engineer biological systems. Mathematical models are incredibly useful across all engineering disciplines, and their utility extends profoundly into synthetic biology. In this field, a mathematical model serves as a formal and crucial link between the initial conceptual design of a biological circuit and its eventual physical realization. It allows researchers to translate abstract ideas into a quantifiable framework. Models enable us to test and optimize various biological designs in silico, which means in the computer, using model-based computer simulations and mathematical analysis. This is done before committing to the more time-consuming and resource-intensive process of creating these circuits in vivo, in living organisms, or in vitro, in a test tube. This predictive power saves significant time and effort. Furthermore, mathematical models help us to generalize experimental data in an understandable and simple manner, allowing us to derive fundamental principles from complex biological observations. They provide a structured way to understand, predict, and ultimately engineer life with greater precision and foresight. When constructing a mathematical model for protein expression, we can consider a biological circuit in terms of its constituent molecules, often called species, and the various processes, or reactions, that link them. The key molecules involved in protein expression typically include a transcription factor, which regulates gene activity, the DNA itself, containing the gene messenger RNA, or mRNA, which carries the genetic code from DNA, and the final protein product. The fundamental processes, or reactions, include gene regulation, which controls when and how much a gene is expressed, transcription, the synthesis of mRNA from DNA, translation, the synthesis of protein from mRNA, degradation, the breakdown of mRNA and protein, and dilution, which refers to the reduction in concentration due to cell growth and division. By breaking down the system into these manageable components and processes, we can begin to build a mathematical representation that captures its essential dynamics. To simplify the conceptualization of these biological processes, we often use compartment diagrams or models. For now, let's focus on mRNA and protein. In a compartment diagram, variables, or species like mRNA and protein are represented as blocks, symbolizing distinct compartments or states within the cell. The processes that transform or move these species, such as transcription, translation, decay, and dilution, are represented as arrows connecting these blocks. For mRNA, transcription leads to its production, while decay and dilution lead to its loss. Similarly, for protein, translation leads to its production from mRNA, and then decay and dilution contribute to its loss. These diagrams provide a clear, visual way to describe the system, making it easier to conceptualize the flow of material and energy within the biological circuit. The mass balance of species forms the cornerstone of mathematical modeling in biological systems. Using a compartment model, we can construct a mathematical representation to describe the change in concentration of each species over time. In general terms, the rate of change of any species' concentration over time is equal to the sum of all rates of its production minus the sum of all rates of its loss. For mRNA, its production occurs through transcription, the process where DNA is read to create an RNA copy. The loss of mRNA happens primarily through two mechanisms: decay, where enzymes like RNase break down the mRNA molecule, and dilution, which occurs as the cell grows and divides, effectively spreading the existing mRNA molecules across a larger volume. Similarly, for protein, its production is achieved through translation, where mRNA instructions are used to synthesize amino acid chains. Protein loss also occurs through decay, catalyzed by proteases that break down protein molecules, and through dilution as the cell increases in size and divides. These rates of concentration change can be precisely captured using ordinary differential equations, providing a dynamic mathematical description of the biological circuit. In the endeavor to simplify biological models, several assumptions are often made because biological systems are incredibly complex. It would be exceedingly difficult to create a truly accurate model without some degree of simplification. These assumptions help to reduce the number of variables and parameters, making the models more tractable. One common type of assumption is the mass action rate assumption. This assumes that the rate of a biological process is proportional to the product of the concentrations of the reactants involved. For example, mRNA decay, which is dependent on ribonucleases, RNase, can be modeled this way. If we assume that the concentration of RNase is constant, a steady-state assumption in itself, then the rate of mRNA decay can be simplified to be proportional only to the concentration of mRNA. The constant of proportionality, K sub m deca sub, is the mRNA decay constant, with units of ones time. This simplification allows us to represent complex enzymatic reactions with simpler mathematical expressions, making the model more manageable. Another simplifying approach is the zero-rate assumption. This assumption is made when the rate of one process is significantly smaller than the rate of other processes. In such cases, the slower process can be considered negligible and effectively ignored in the model without substantially impacting its accuracy. For instance, in our example, if the mRNA dilution rate, with a half-life of approximately 30 minutes, is much lower than the mRNA decay rate, with a half-life of about 5 minutes, we can assume that the total mRNA loss rate is essentially dominated by the decay rate. Similarly, if the protein decay rate, with a half-life of 2 to 3 hours, is much lower than the protein dilution rate, with a half-life of about 30 minutes, we can assume that protein loss is primarily driven by dilution. By applying this zero-rate assumption, we can simplify our ordinary differential equations, removing terms that represent the negligible processes and focusing on the dominant mechanisms of change. Following the zero-rate assumption, we can simplify our mathematical description significantly. Therefore, for mRNA, we can confidently say that its loss is primarily dominated by decay, and the term for dilution can be omitted. Similarly, for protein, its loss is dominated by dilution, allowing us to simplify its rate equation by removing the decay term. This streamlined approach makes the mathematical models more manageable while still capturing the most significant biological dynamics. For mRNA, the rate of change becomes the rate of transcription minus the rate of mRNA decay. For protein, the rate of change is the rate of translation minus the rate of protein dilution. These simplified ordinary differential equations provide a clearer and more concise representation of the system's behavior. A third important simplification is the constant rate assumption. This assumption is applied when certain processes occur at a rate that can be considered constant over the relevant time scale of the model. For example, some housekeeping genes within a cell are constitutively expressed, meaning they are transcribed at an almost constant rate, regardless of external conditions. For these genes, the rate of transcription can be represented simply as a constant value, K sub TRSC sub, which has units of concentration per unit time. We can directly enter these constants as parameters into our model. The values for these constants are typically determined through experimental measurements, ensuring that the model reflects observed biological reality. This assumption simplifies the mathematical equations by replacing dynamic rate expressions with fixed values, making the model more straightforward to solve and analyze. Applying the constant rate assumption further simplifies our mathematical models. For example, if a gene is constitutively expressed, meaning it is always on and producing its product at a steady pace, we can assume it has a constant transcription rate. So, the rate of mRNA change, which was initially expressed as the rate of transcription minus the rate of mRNA decay, can now be simplified. It becomes the constant transcription rate, K sub TRSC sub, minus the rate of mRNA decay. This substitution streamlines the equation, making it easier to work with while still accurately representing the biological process under the steady, constant conditions assumed. The fourth type of assumption we often employ is the steady-state assumption. This is a powerful simplification that assumes the net change in concentration of a particular species over time is zero, meaning its concentration remains constant. In other words, the rates of production and loss for that species are balanced. This assumption is usually applied to intermediate species in a reaction pathway, species that have much quicker kinetics, meaning they are produced and degraded very rapidly, or to housekeeping species whose concentrations are tightly regulated and maintained at stable levels within the cell. For example, if there's a perturbation to a biological system, the concentration of mRNA often recovers to a stable level much quicker than the concentration of its corresponding protein. In such a scenario, we can make a steady-state assumption for mRNA. This means that at steady state, the rate of mRNA transcription equals the rate of mRNA loss. This simplification helps to reduce the number of dynamic equations in a model, allowing researchers to focus on the slower, rate-limiting steps and the overall system behavior, rather than the rapid fluctuations of intermediate species. Let's now consider the concept of parts in synthetic biology, which are the foundational DNA sequences encoding specific biological functions. These parts are the basic building blocks from which more complex biological systems are constructed. Some common biological parts include promoters, ribosome binding sites, protein coding sequences, and terminators. Each plays a distinct and crucial role. Promoters are DNA sequences that initiate transcription, recruiting RNA polymerase to begin copying the gene into messenger RNA. Ribosome binding sites initiate translation, attracting ribosomes to synthesize protein from the mRNA. Protein coding sequences encode for the actual protein, containing the instructions for its amino acid sequence. And terminators signal the end of transcription, causing RNA polymerase to detach from the DNA. These individual, well-defined parts are like standardized components in engineering, allowing synthetic biologists to assemble functional biological devices in a modular fashion. Promoters are absolutely essential parts in any biological circuit, acting like the on switch for a gene. A promoter is a sequence of DNA that specifically recruits RNA polymerase, along with other accessory proteins, to prime the transcription of messenger RNA, or mRNA. Without a promoter, a gene cannot be effectively read and converted into its protein product. There are two common types of promoters. First, constitutive promoters are not regulated by any DNA-binding proteins. They are essentially always on, driving gene expression at a constant level. They provide a continuous baseline of activity. Second, regulated promoters often contain additional DNA sequences known as operators. These operators allow other protein factors, called repressors or activators, to bind to the DNA. Depending on whether a repressor or activator is present, these factors can modulate, or control, the transcription rate, either turning the promoter off or increasing its activity. This ability to control gene expression is fundamental to building sophisticated biological circuits. Constitutive promoters, as their name suggests, provide a constant level of expression for a protein of interest within a biological system. They are always active, driving the transcription of genes without requiring any specific regulatory signals. The beauty of constitutive promoters lies in their inherent simplicity and predictability. However, even within constitutive promoters, there is room for precise engineering. By carefully varying the specific sequence of the DNA that makes up the RNA polymerase binding site within the promoter, it's possible to adjust its strength. A stronger binding site will recruit RNA polymerase more efficiently, leading to an increased rate of mRNA production. Conversely, a weaker binding site will result in a lower rate. This ability to modulate the promoter's strength allows synthetic biologists to fine-tune the steady-state concentration of the protein produced. Essentially, you can build a constitutive promoter that is always on, but you can also design it to be always on, but at a low, medium, or high level, providing an important knob for controlling gene expression without external induction. Regulated promoters offer a more sophisticated level of control over gene expression. These promoters can be either positively regulated or negatively regulated. In positively regulated promoters, specific factors binding to the DNA actually increase the transcription rate, acting like an accelerator for gene expression. In negatively regulated promoters, the binding of certain factors to the DNA decreases the transcription rate, effectively acting as a brake. In many biological systems, negative regulators are naturally present within the cell. Under normal conditions, these regulators bind to the promoter, preventing transcription. In such cases, the default state of the promoter is off. However, these promoters can be induced or turned on by specific molecules or by changes in environmental conditions, such as temperature or pH. For instance, the introduction of a particular molecule might cause the repressor to detach from the DNA, thereby allowing transcription to proceed. This inducible nature is key to building responsive and dynamic biological circuits, allowing precise control over when and where a gene is expressed. The operator is a crucial component in the regulation of gene expression, particularly for regulated promoters. It is a specific DNA sequence that acts as a binding site for regulatory factors. These factors can be either repressors, which negatively regulate the promoter by inhibiting transcription, or activators, which positively regulate the promoter by enhancing transcription. When a repressor binds to the operator, it physically blocks RNA polymerase from initiating transcription, effectively turning the gene off. Conversely, when an activator binds, it can help recruit RNA polymerase or facilitate its activity, thereby turning the gene on or increasing its expression. In synthetic biology, the operator is often not considered a separate part but rather an integral component of the promoter itself. The promoter-operator set is then characterized together as a single functional unit. The precise DNA sequence of the operator can profoundly affect the binding strength of the repressor or activator. Even the DNA sequence located proximal, or close, to the operator can influence protein-DNA interactions, further modulating the regulatory effect. This intricate interplay between the operator and its binding factors allows for fine-tuned control over gene expression, which is essential for designing complex biological circuits. Another vital component in the intricate process of protein synthesis is the ribosome binding site, or RBS. This is a specific sequence of DNA that, once transcribed into messenger RNA, serves as a crucial recognition point. Its primary role is to recruit ribosomes, the cellular machinery responsible for protein synthesis, to the mRNA molecule. This recruitment is the very first step in the initiation of protein translation. In prokaryotic organisms, such as bacteria, this sequence is well known as the Shine-Dalgarno sequence. In eukaryotic organisms, which include plants, animals, and fungi, it is known as the Kozak consensus sequence. Like promoters and operators, the precise sequence of the ribosome binding site profoundly affects the strength of ribosome binding. This, in turn, directly influences the amount of protein that is ultimately synthesized from that mRNA molecule. By engineering the RBS, synthetic biologists can fine-tune the rate of translation, allowing them to control the final output of protein in a designed biological circuit. The protein coding sequence, or CDS, is the core of any gene, holding the instructions for building a protein. It is the specific DNA sequence that directly encodes for a protein, excluding non-coding regions like introns found in eukaryotes. The CDS begins with a start codon, typically ATG, which signals the initiation of protein translation. It then consists of a series of codons, each 3 nucleotides long, which specify the sequence of amino acids that will make up the protein. The sequence ends with one of 3 stop codons, TAA, TAG, or TGA, which signal the termination of translation. In between the start and stop codons, the codons denote the specific amino acids that will be assembled to form the protein. This precise sequence is the fundamental blueprint that determines the identity and function of the protein product. Within protein coding sequences, reporters are a particularly useful category. These are genes that encode proteins designed to serve as clear readouts of dynamic biological events. Typically, a reporter protein produces a visual or fluorescent output, making it easy for researchers to observe and quantify gene expression or other cellular activities. A classic example is the green fluorescent protein, or GFP. When a gene is linked to GFP, and that gene is expressed, the cells produce GFP, which then fluoresces brightly under specific light, showing a clear output. This allows scientists to visually track gene activity, protein localization, or the success of a genetic circuit, making complex biological processes readily observable and measurable. Reporters are indispensable tools for debugging, optimizing, and demonstrating the function of engineered biological systems. Another critical class of protein coding sequences are regulators. These proteins bind to specific DNA sequences, particularly the operator region of promoters, to control gene expression. They can either repress or turn off transcription, or induce or turn on transcription. Transcriptional regulators are essentially the switches of a biological circuit. Regulators can also contain binding sites for small molecules. These small molecules act as signals, influencing the regulator's ability to bind DNA. For instance, a small molecule might bind to a repressor, causing it to detach from the operator and thereby switching transcription on. Conversely, another small molecule might bind to an activator, preventing it from binding to DNA and switching transcription off. These intricate molecular switches allow synthetic biologists to design genetic circuits that respond to specific environmental cues or internal signals, creating sophisticated control over cellular behavior. Beyond reporters and regulators, protein coding sequences also include enzymes, which are vital for performing specific chemical reactions essential for building and maintaining biological systems. Enzymes can be involved in biosynthesis, the production of new molecules or metabolites, or in their degradation. They play a role in DNA modification, such as restriction, which involves cutting DNA, or ligation, which involves joining DNA fragments. Proteases are specific types of enzymes that cleave or degrade other proteins, controlling their lifespan and function. Additionally, enzymes are crucial for post-translational modifications, which are changes made to proteins after they have been translated. These modifications, such as methylation, phosphorylation, or glycosylation, can alter a protein's activity, localization, or stability, further expanding the functional complexity of biological systems. Other essential proteins encoded by protein coding sequences include membrane proteins. These proteins are embedded within or associated with cell membranes and perform a diverse array of functions. Examples include surface display peptides, which can present specific molecules on the cell surface, transporters, which move substances across cell membranes, channels, which create pores for ion flow, and pumps, which actively transport molecules against their concentration gradients. Receptor and ligands are another important pair. Receptors are proteins that bind to specific signaling molecules, or ligands, initiating a cellular response, effectively serving as receiving and signaling molecules. Lastly, lysis proteins are a group of proteins involved in killing cells, often by disrupting their cell membranes. These are sometimes used in synthetic biology for applications where programmed cell death is desired, for example, to contain engineered organisms or to release specific products. Terminators are the final essential parts in a transcriptional unit, acting as the stopped signal for RNA polymerase. A terminator is a sequence of DNA that, when transcribed into RNA, causes the RNA polymerase to cease transcription and disengage from the DNA template. It is strategically placed past the 3' end of a protein coding sequence, ensuring that the entire gene has been transcribed. In prokaryotes, terminators often consist of palindromic DNA sequences. When these sequences are transcribed into RNA, they form strong secondary structures, typically a hairpin loop, due to internal base pairing. This hairpin structure then helps to weaken the interaction between the RNA polymerase and the DNA strand being copied, causing the polymerase to pause and eventually fall off. This elegant mechanism ensures that transcription ends precisely where it should, preventing the production of unnecessarily long or non-functional RNA molecules. Terminators are crucial for defining the boundaries of gene expression and are carefully chosen in synthetic biology to ensure predictable and efficient operation of engineered circuits. Moving up the levels of abstraction, device-level design involves assembling these individual parts into functional devices that perform simple, defined biological functions. A device is a modular combination of parts that executes a specific operation, much like an electronic circuit combines resistors and capacitors to perform a task. For example, a basic genetic device might consist of 4 parts: a promoter, a ribosome binding site, RBS, a protein coding sequence, CDS, and a terminator. This device would constitutively produce the protein encoded by the CDS. By combining these well-characterized parts, synthetic biologists can create increasingly complex functions, building from basic genetic switches to more elaborate regulatory networks, much like building with specialized blocks to create a machine. This modular approach allows for systematic design, testing, and troubleshooting of biological circuits. One common and powerful type of device that synthetic biologists aim to build are biological logic gates. Logic gates, in the realm of electronics, are the fundamental building blocks of digital systems, enabling computers to perform complex operations. These gates perform logical operations based on Boolean algebra, taking one or more binary inputs and producing a single binary output. Just as electronic engineers build circuits with or not gates, synthetic biologists are now working to construct biological devices that mimic these same functions within living cells. By engineering cells to respond to specific molecular inputs and produce distinct molecular outputs, we can essentially program biological systems to perform computations, detect combinations of signals, and make decisions, paving the way for advanced diagnostics, therapeutics, and intelligent biomanufacturing. Boolean logic forms the mathematical foundation for these biological logic gates. It is a form of algebra where variables can only have 2 values: true, often represented by 1, or false, represented by 0. This binary system is what underpins all digital computation. The basic operators of Boolean logic, which are essential for building any complex logical function, are and or, and not. Let's gently review these basic operators. An AND gate will only produce a true 1 output if all of its inputs are true 1. If any input is false 0, the output will be false 0. An OR gate, on the other hand, will produce a true 1 output if at least one of its inputs is true 1. It will only produce a false 0 output if all of its inputs are false 0. Finally, a NOT gate, also known as an inverter, takes a single input and inverts it. If the input is true 1, the output is false 0, and if the input is false 0, the output is true 1. Understanding these fundamental logical operations is key to comprehending how biological systems can be designed to process information. Negated gates are formed by combining a NOT gate with another logic gate, which effectively inverts the function of that gate. This creates new logical operations that are also crucial in designing complex circuits. For example, combining a NOT gate with an AND gate results in a NAND gate. A NAND gate will produce a true 1 output if any of its inputs are false 0, and only a false 0 output if all of its inputs are true 1. It's the inverse of an AND gate. Similarly, combining a NOT gate with an OR gate produces a NOR gate. A NOR gate will yield a true 1 output only if all of its inputs are false 0. If any input is true 1, the output will be false 0. This is the inverse of an OR gate. These negated gates expand the toolkit for building sophisticated biological logic, allowing for a wider range of conditional responses within engineered cells. Biological logic devices, or logic gates, are a fascinating application of synthetic biology. In this field, we can construct devices that exhibit the functions of these Boolean logic gates within living cells. Imagine you want to create a device that reports a signal, perhaps through green fluorescent protein or GFP, only when a specific protein, say TETR, is absent. This would be a biological NOT gate, or an inverter. One possible way to construct this inverter is to identify an operator sequence that is repressed by TETR. You would then connect this operator and a suitable promoter to a GFP coding sequence. In this design, when the repressor protein TETR is present, it binds to the operator, preventing the transcription of the GFP gene, resulting in no fluorescence, output 0. However, when TETR is absent, it cannot bind to the operator, allowing the GFP gene to be transcribed and translated, leading to fluorescence, output 1. The associated truth table for this biological NOT gate would show, if TETR is 0, absent, GFP is 1, fluorescent, if TETR is 1, present, GFP is 0, not fluorescent. This simple circuit demonstrates how biological components can be engineered to perform fundamental logical operations. Building on the concept of logic gates, we move to system-level design. This stage encompasses two critical aspects: defining the overall system behavior or function, and selecting the appropriate chassis or host organism. The first step in system-level design is always to clearly define the desired behavior or function of the entire system. What problem are we trying to solve, and what specific output do we want the engineered biological system to produce under certain conditions? Consider a heavy metal sensing example. Our desired function might be, if zinc and iron are both present, or if lead is present, then the system should produce an output, specifically a color change, meaning color should be produced. If none of these conditions are met, there should be no output, no color should be produced. This clearly defines the complex logical function the system needs to perform. Once this system-level function is clearly articulated, we can then begin to break it down into the individual devices and parts needed to achieve this sophisticated behavior. This top-down approach ensures that the entire engineering effort is focused on meeting the ultimate goal. A second crucial aspect of system-level design is determining which host organism, or chassis, you will use to run your genetic program. The chassis provides the underlying biological machinery necessary to transcribe and translate the engineered genetic system, as well as supplying any enzymatic substrates required for the desired reactions. Essentially, it's the living platform within which your synthetic circuit will operate. You can use many different organisms as a chassis, including bacteria, yeast, viruses, or even mammalian cells, each offering different advantages and complexities. Alternatively, the chassis can even be abiotic, meaning it's not a living organism but rather a cell-free system that provides only the necessary biochemical components for in vitro transcription and translation. However, the most common chassis organisms used in synthetic biology are the bacterium E. coli and the yeast Saccharomyces cerevisiae, due to their well-understood genetics, ease of manipulation, and rapid growth rates. The choice of chassis depends heavily on the complexity of the desired biological circuit and its intended application. Some synthetic biologists are exploring even more advanced concepts, such as minimal cells or synthetic cells. This involves engineering cells to be streamlined for a single, specific task, making the system more efficient and requiring fewer resources than a complex natural cell. The idea is to strip down a cell to its most basic, essential components and then build it back up with only the genetic circuits necessary for its engineered function. This bottom-up approach to cell construction means that the engineered cells could be made dependent on specific external conditions. This could, for instance, prevent them from living in undesired environments, offering a containment strategy for environmental applications. A significant advantage is that engineered cells may behave more predictably compared to natural cells, which often have complex, redundant, and highly interconnected regulatory networks that can make their behavior difficult to predict. This pursuit of minimal and synthetic cells represents a frontier in synthetic biology, aiming for greater control and predictability in engineered biological systems. The biological system design process often follows a hierarchical approach, moving from the broad concept to the minute details. When designing a biological system, we typically start at the highest level, the system design level. At this stage, the focus is on understanding the overall function of the system that can address the problem we are trying to solve. For example, if the problem is heavy metal contaminated water in developing countries, the system might be a cell that produces a color response when lead and iron, or mercury, are present, allowing differentiation between contaminated and clean water. Once the system's function is defined, we then move down to the device design level. Here, we decide what specific devices will be needed to construct the system. A device converts some input into a specific output. For our heavy metal example, we might need devices that sense iron, lead, and zinc, and a device that generates a color change. The input for the heavy metal sensing devices would be the heavy metals complexed with their binding proteins, and the output would be a protein that serves as input for the color-generating device. The output of the color-generating device is a chromoprotein, producing the visual signal. Next, we descend to the parts design level. This involves choosing the individual biological parts, such as promoters, ribosome binding sites, and coding sequences, that will be used to build each device. For the heavy metal sensors, this might involve promoters sensitive to specific metals and coding sequences for metal binding proteins. For the color-generating device, it would involve a coding sequence for a chromoprotein. Finally, we reach the DNA-level design, where we find or create the actual DNA sequences that will make up each part. This involves searching existing registries of standard biological parts or synthesizing new sequences based on literature. This methodical descent through levels of abstraction ensures that the complex task of designing a biological system is broken down into manageable, interconnected steps. Challenges with biological circuits are numerous and highlight the differences between engineering living systems and traditional electrical circuits. Electrical circuits benefit from explicit physical wiring, where connections are clearly defined. They operate with rapid signals, allowing for near-instantaneous responses. The components are typically identical devices, ensuring predictable behavior across multiple units. Electrical engineering also has established designs and many formal languages for describing and simulating circuits. Biological circuits, in contrast, face more complex challenges. They often involve implicit chemical wiring, where interactions occur through molecular diffusion and binding rather than fixed physical connections. This leads to differing signal rates, which can be slower and more variable. Biological devices are inherently unique, exhibiting variability even between genetically identical cells due to stochastic biological processes. The field is still in the process of developing designs and currently lacks a universally accepted formal language for describing and predicting the behavior of complex biological systems with the same precision as electrical circuits. These challenges underscore the unique complexities and exciting frontiers of synthetic biology. Beyond the technical challenges, there are other important considerations for potential applications of synthetic biology. An application should ideally be general, meaning it can be applied to a wide range of situations or users, rather than being limited to custom solutions. This broad applicability helps justify the significant developmental cost often associated with novel biotechnologies. An application should also be compatible with its chosen biological chassis. For instance, if designing a system for exothermic, heat-generating applications, a thermophile, a heat-loving bacterium, would be a suitable chassis. Crucially, any application must be designed to be contained, to avoid contamination of natural environments or other biological systems. This often involves engineering kill switches or other safeguards to prevent the spread of engineered organisms. Finally, and perhaps most importantly, public acceptance or opposition plays a vital role. The ethical and societal implications of genetic engineering mean that public understanding, engagement, and trust are essential for the successful and responsible deployment of any new biological application. These considerations collectively shape the responsible development and integration of synthetic biology into society. As we gently drift towards the conclusion of our exploration, let us take a moment to reflect on the profound journey we have undertaken through the landscape of genetic engineering and synthetic biology. We have touched upon its history, from the ethical lessons of the Nuremberg Code and the Tuskegee study to the guiding principles of the Belmont Report. We have softly examined the foundational techniques, like PCR, restriction, and ligation, the very molecular tools that allow us to interact with life's blueprint. We've considered the modular nature of synthetic biology, building complex functions from simple parts, much like constructing with biological Legos. We explored how these parts can form biological logic gates, allowing cells to make decisions based on inputs, a truly remarkable feat of bioengineering. The immense power and diverse applications of this field, from targeted therapies and sustainable manufacturing to environmental remediation, paint a picture of a future both promising and awe-inspiring. Yet, with every whisper of possibility, we are reminded of the deep ethical questions that accompany such power. The debates around playing God, the distinction between living organisms and engineered machines, and the profound responsibilities of managing potentially dual-use knowledge continue to evolve. These are not simple questions with easy answers, but ongoing conversations that demand our thoughtful and compassionate engagement. Genetic engineering is not just a scientific discipline, it is a profound reflection of our human ingenuity and our ongoing quest to understand and shape the world around us. It is a field that constantly challenges us to balance innovation with responsibility, ambition with caution, and knowledge with wisdom. As the boundaries of what is possible continue to expand, so too must our commitment to ethical reflection and societal well-being. May your mind now rest peacefully, carrying a gentle understanding of these intricate and important concepts. Good night.